डॉक्टर बर्मन, हेलो।
यस सर।
बर्मन, क्या देरी है शुरू होने में?
सर वो डॉक्टर BS ढिलों सर अभी ज्वाइन कर रहे हैं, उनके लिए वेट किया जा रहा है सर।
अच्छा, जो नहीं किया उन्होंने।
यस सर।
No, he is joined, sir.
ठीक है, वेरी नाइस। चलिए, कितने हो गए दाऊजा जी?
10 हो गए सर।
अच्छा, 14 बचे हैं अभी।
टोटल 22 थे सर।
अच्छा, 22 है, 12 बचे हैं अभी। ठीक है, इट मीन्स 3 hours will require.
Can we start now, sir?
Dr. Saab joined?
Yeah, yes, sir.
Okay. So, sir, I think when we finished the last presentation, your remarks were due, comments if any. So, I will invite you for that first before we go to the next presentation.
Thank you, Dr. Saab. I will continue what Dr. Joshi said. Beautiful photographs, enjoyable slides, wonderful. And then thanks to Komal as well as Dr. Shishan, her advisor.
Yes, sir.
Yes, sir.
Yeah, for taking this. I mean, this taxonomic tribe is disappearing, whether
Yes,
it's plant taxonomy or in geology or in botany. So doing something taxonomy, I felt very happy that some efforts are going, and I was pleasantly surprised that so much diversity in Pusa campus itself. Maybe
Yes,
we had
sir.
written cross. So at the moment, nothing more because I don't know much about taxonomy. इतने बड़े-बड़े नाम हैं, समझ तो आता नहीं है कुछ भी। तो ये डॉक्टर बांसल साहब को ज्यादा आता होगा या डॉक्टर जोशी जी को। ये BHU के स्टूडेंट, हम तो एग्रीकल्चर यूनिवर्सिटी के स्टूडेंट हैं थ्रू आउट। BHU is multi-faculty university, and I think Dr. Bansal, you are from Delhi University, BSc.
No, BSc I am from Punjab University, Punjab.
So even then, that is multi-faculty university. Punjab University, Dr. P.N. Mehra was in the selection committee of Dr. Swaminathan. P.N. Mehra
Right.
was in the selection committee of Dr. Swaminathan, director of IRA. Anyway, so it was really a very pleasing presentation.
Thank you so
And
much.
all
sir.
the best, Bete. All the best.
Thank you, sir.
So let
Thank
us
you,
know.
All right, Dr. Dahuja, please go ahead to the next presentation.
Okay. Sir, as you know that we missed one presentation by Miss Pratiksha Suba because recording was not good because of the bad weather at her end. So we may allow her to present her study through the PowerPoint presentation.
Please.
So,
Please.
okay. So with this, I invite Pratiksha Suba to please share her slide and start the presentation.
So is it visible, sir?
You can put it in the
Yeah.
slideshow mode.
Yes, sir. I'm very sorry for inconvenience, sir.
No, we are sorry. Not why you should be sorry. COVID, you didn't bring COVID. Please move on. Wonderful.
A very good afternoon to everyone. My name is Pratiksha Subba. I'm
You are
from
not,
division of.
Pratiksha, you are not in slideshow. Can you put in slideshow?
Sir, I have kept it in slideshow, sir. Now.
Okay,
But at
so then
overall-
it is not here at this end. There is some, bug related issues, but you make it sure that when you change your slide, then you have to click on the next slide. Just change the slide and see if the slides are moving.
Sir, is it moving, sir?
No, not at this end. So you have to go, in fact, in this, you keep your presentation also in the slide, working mode, and then go slide by slide.
Sir, actually she has a lot of animations in each slide, so that will not work if she is not in slideshow mode.
Then ask her to send the slide to you by mail, and
Yes,
you
sir.
share from here. We
Okay,
will
sir.
take another presentation. Yeah.
Okay, sir.
Okay, sir.
Sorry, Pratiksha. Yeah, it's not.
Sir,
moving.
sir, I'm so sorry, sir.
Okay, please
No,
unshare and
Bete, you need not be sorry.
send it.
You keep your
Send-
spirits up.
Yeah.
Send your presentation to Burman immediately. Yeah. We go to the next one.
Okay. The next presentation is by Miss Pooja from Division of Environmental Science.
I extend my warm welcome to everyone to my IRI Merit Medal Presentation 2022. Let me introduce myself. I'm Pooja Ella from Division of Environment Sciences, and my chairperson is Dr. Renu Singh. I would like to start my presentation by quoting the inspiring words by Kofi Annan, the 7th Secretary General of United Nations. I quote, "Our biggest challenge in this new century is to take an idea that seems abstract, sustainable development, and turn it into reality for all the world's people." The global population is growing exponentially and has reached 7.9 billion because of increase in food production. The sole reason behind this is synthetic nitrogen fertilizer. It alone supports 3.5 billion. Otherwise, the world's population would be somewhere around 3.5 to 4 billion. Agriculture alone is responsible for 80 to 95% of total ammonia emissions in the atmosphere. Fertilization with mineral nitrogen, especially urea, is responsible for 19 to 20.3% of total ammonia emitted from agriculture. Let us understand the fate of synthetic nitrogen fertilizers, which are given as inputs and also lost in many ways. Taking urea as example, when we apply urea into the field, only part of it is uptaken by the crop and the rest is lost. In that, the major pathway of gaseous emission is through ammonia volatilization. Up to 80% of applied urea is lost as ammonia gas, that is conducive to low nitrogen use efficiency and indirect emission of nitrous oxide. Then, up to 20% is lost as N2 and 3% is lost as N2O through denitrification process. Up to 25% is lost as nitrate through leaching to the underground, contaminating the groundwater. With this background, I took up a research on reducing ammonia volatilization using organic and inorganic amendments in wheat. The objective of my study were: the first objective was to study the effect of organic and inorganic amendments on ammonia volatilization, and my second objective was to evaluate the effect of organic and inorganic amendments on nitrogen use efficiency and productivity of wheat. The details of my experiment are like this. My study was taken up in MB3C plot at IRI with a wheat variety HT2697 in the plot size 4 by 3 meter square, each for 10 treatments with 3 replications in randomized block design. For our experiment, we have picked up some plant or microbe-based amendments like WAM, Azotobacter, garlic powder, linseed oil, and Pongamia oil, and also we have taken chemical inhibitors like NBPT, that is N-butyl thiophosphoric triamide, hydroquinone, and also calcium carbide. The treatments were T1, control with no recommended dosage of nitrogen, T2, RDN at the rate of 150 kg per hectare, T3, RDN plus NBPT at the rate of 0.05% weight by weight basis, T4, RDN plus hydroquinone at the rate of 0.3%, T5, RDN plus calcium carbide at the rate of 1%, T6, RDN plus WAM at the rate of 10 kg per hectare, T7, RDN plus Azotobacter at the rate of 50 gram per kg seeds, T8, RDN plus garlic powder at the rate of 0.8%, and T9, RDN plus linseed oil at the rate of 0.06%, T10, RDN plus Pongamia oil at the rate of 0.06% on weight by weight basis. Around 14 different plant and soil parameters were analyzed at different stages of crop growth using different methodologies and instruments. The initial soil properties like soil pH, EC, and available NPK, ammonical N, and nitrate N was analyzed before sowing the crop. Total nitrogen content and available N was found to be low. Ammonia fertilized from the field was estimated using forced air draft method, where a closed chamber measuring 20 by 20 by 50 cm made up of 6 mm acrylic sheets were placed in the field. Volatilized ammonia gas from the soil surface under different treatments were collected in the 2% boric acid traps with mixer indicator using vacuum pump. These traps are changed for every 24 hours, then titrated against 0.02 normal sulfuric acid and further calculated using equation. This slide shows glimpses of my research work and different stages of my research work. The total N content and available N content in the soil when analyzed using Jaldal method after harvesting of the crop found to be highest in Azotobacter treated plots. The increase in the N content is due to the addition of biofertilizers as amendments. This in turn has increased the residual N, which will be reducing the N fertilizer application for the next crop. The treatments with NBPT performed well in reducing ammonia losses by 40% due to strong inhibition of urease activity. Among plant-based amendments, T8 treated with garlic powder reduced ammonia volatilization by 17% when compared to control due to the allicin content present in it, which acts as a bioinhibitor of urease activity. The temporal graph showed two peaks of ammonia flux in the entire cropping season, depicting that the major losses were observed in the initial 5 to 6 days after the fertilization. The available N forms, particularly ammonical N and nitrate N, found to be decreasing from the tillering stage to grain filling stage. In all the stages, the highest ammonical N and nitrate N were observed in NBPT treated plots due to the slowing down of urease activity and enhancing the accumulation of these ions. The soil biomass carbon and soil biomass nitrogen was also found high in Azotobacter treated plots, as the microorganisms helped in decomposition of organic matter, hence releasing the essential nutrients. And the activity of urease enzyme was also found low in NBPT treated plots. The highest nitrogen content in both straw and grain was found in T3, that is NBPT treated plot. Again, this might be due to the improved nitrogen availability due to the reduction of N losses and also better nitrogen use efficiency. To quote few important findings of my study, NBPT, that is N-butyl thiophosphoric triamide, performed well by reducing the ammonia losses by 40% when compared to control. And among plant-based treatments, garlic powder treated urea showed better results by decreasing the ammonia losses by 17% when compared to control. The available N forms, majorly ammonical N and nitrate N, found high in NBPT amended treatments for plant uptake. And the highest grain yield was found in treatment treated with Azotobacter. And the highest N content in the grains was observed in NBPT treated plots. The future perspectives of my study are: plant-based nitrogen inhibitors can be developed as economical and eco-friendly alternative strategy to reduce nitrogen losses and also to increase the yield. The impact of natural nitrogen inhibitors and cost-benefit analysis for different crops can be studied. My academic achievements are: I feel fortunate to be one among 44 graduates and postgraduates from all over India to witness 72nd Republic Day Parade from Prime Minister Box, and also felicitated with a certificate of appreciation by the former Minister of Education, Shri Ramesh Pokhriyal sir. I have published one conference paper at Xrd Procedia 2020 and one review paper in Annals of Agricultural Research and one abstract in ISA Savanna 2021 and one popular article in International Monthly E-Magazine. A book chapter on lignocellulosic biomass is submitted to Springer Nature and is being accepted. One research paper is being accepted in Annals of Agricultural Research. A research paper on my thesis is submitted and under review in Journal of Agricultural Physics. And I have secured ICR SRF rank 3 in Environmental Science subject. And I have created 2 career guidance YouTube videos for environmental science students. I have secured Best Paper Award, Best Oral Presentation Award in international conferences, and Best Article Award from Aggregate Magazine, and I have also presented poster in 6th National Youth Convention. I have work experience at Center for Capacity Building at Environmental Management Policy Research Institute, Bengaluru, and I was also part of last year's Convocation Choir Group. These are few certificate of participation in international and national conferences and webinars. At last, I would like to acknowledge my chairman, members of advisory committee, seniors, friends, and lab members, a hidden professor of Division of Environment Science, Dean, Director, PG School, IRA. Thank you.
ಥ್ಯಾಂಕ್ ಯು ಅಂತ ಬಂತ್ಲೆ ಮುಗೀತು
I will invite Dr. Joshi to please start the interaction with the student.
Thank you very much. So the name was Pooja, if I'm
Yes.
correct.
Yes, sir. Yes.
Yes, sir.
Okay, Pooja ji, very good. I think theme is very attractive because the world has to become sustainable. But the world is changing towards nanoscience. So did you include this nanoscience in some way here, or do you see in future when that will come into the work you are doing, that kind of work? Thank you.
Yes, sir. Thank you, sir. Yes, sir. Surely, that is a great idea, sir. Definitely, if I work, definitely I will work on this in my PhD, sir. That's a great idea, sir.
Okay, thank you.
May I now invite Dr. Bansal for the interaction purposes?
Okay, I thought before me, Doctor. Anyway, Pooja, I want to know,
Yes, sir.
of course, this was a standard experiment. You had all the treatments and all, both in organic interventions, to really see how urea can be utilized more efficiently, right? Or how can we increase nitrogen use efficiency in a sustainable way? But also I wonder, how about other environmental parameters? For example, particularly cultural practices, right, in wheat field. I think irrigation has a lot of interaction, I would say, particularly when we talk of urea application and whatever the effect it has on the environment. I think irrigation has also a role to play. So I wonder whether soil moisture content and also up to what depth of the soil, within the rhizosphere, you were able to see this effect of various treatments, with regard to the objectives you had laid down with you, whether that was a part of your discussion anytime in your thesis.
Yes, sir. We have discussed on this issue. And obviously, that is the major factor, one of the major factor which affects N losses. And we have taken we have taken the soil samples up to 15 centimeters, and we have not analyzed any soil moisture content, sir.
Because what makes this nitrates leach down to the soil and all that, it
Yes.
is, so I think that point should have been taken into account while, if not as particular parameter of your research, but as one of the factors important, I believe. Yeah.
Yes.
And next is that when you apply these various interventions, so at what depth were they, or were they placed, and how were they, or they were applied? If I missed that point, I believe, during your methodology, can you please tell us?
Sir, it is applied 2 to 3 centimeters below the soil before the sowing of the crop, and after we have just broadcasted, sir, in second split.
Okay, as of now, thank you. Thank you.
Thank you, sir.
Thank you, sir. And Dr. Patra, please.
Yes, Ms. Pooja.
Yes, sir.
Sir, you are on mute.
Okay, automatically it happened. Yes, my questions, what was your hypothesis for taking calcium carbide as well as garlic powder, particularly the calcium carbide in mid 80s? There are good studies, at IRI in the Division of Soil Science. So that was coated calcium carbide, not calcium carbide, because calcium carbide, it highly reactive. When you put it, immediately acetylene gas will be released and may not be effective. So there is already, and that has been patented also, and the contribution of the Division of Soil Science and Chemistry, coated calcium carbide. I do not know you have used coated or uncoated one. And regarding the garlic powder, there may be reasons you have taken. So this is one. Another one is, I saw your results that when you give the recommended dose of nitrogen plus NBPT, there was reduction of ammonia losses by 40% or so, 40%. But when the the yields which you obtained that is higher in recommended dose of nitrogen and application of azotobacter. So what may be the reasons? Because azotobacter may fix some nitrogens, which is a possibility is there, but also substantial amount of ammonia loss is also reduced. So this is, please clarify.
Yes, sir. Thank you, sir. Sir, to answer your first question, I would like, we put in mind that we should consider urease inhibitors or chemical amendments, which means we have taken NBPT as reference, and we search for inhibitors which are of low cost and which can give better results. And garlic, that is a new approach. Even though no field studies have been taken in applying garlic powder as an amendment in order to reduce ammonia volatilization. So if it could give us better results, we can go for alternatives. Instead of only neem coated urea, we can go for garlic powder coated urea, which will reduce the losses and also increase the nitrogen use efficiency. And to answer your second question, sir, even though NBPT, the yields are very means comparatively less than the azotobacter, because applying the biofertilizers like azotobacter, they have good effect in root proliferation. So when the root growth is very good, the absorption of the nutrients will be higher. So the intake and nitrogen use efficiency is higher in azotobacter treated plots. So in turn, it has increased the yield bit, sir.
Right. Class number.
Okay. Thank you. Thank you, Ms. Pooja.
Thank you, sir.
Dr. Dahuja,
Yes, sir.
may I have?
Yeah, please go ahead, sir.
Okay, Pooja, on the basis of the findings, you have recommended garlic powder.
Yes, sir.
Have you worked out the additional cost involved in putting this garlic powder? On which basis you can say that, okay, it is economical?
Yes, sir. Sir, we bought, when we bought garlic, we bought fresh garlic and then we dried and then we powdered. For 1 kg, about 250 rupees I paid. So if we extract and around 1:10% on basis of 1% dry weight we will get. So it is much economical when I compare to NBPT, and also it is giving around, it is decreasing the ammonia volatilization by 17% when compared to control. So I can suggest that compared to all other plant-based amendments I have used, so garlic is better. And if we work on garlic and we can come out with some products and we can get a solution.
It's okay, but when you add garlic powder cost, then you should also add some additional labor which is required to put it into it. Okay.
Yes, sir.
Okay, thank you.
Thank you, sir. So may I now invite comments from the chairperson?
Okay, Dr. Sir, thank you. So, Pooja, congratulations for working on a very pertinent, very important issue. Use of NP, use efficiency of nitrogen phosphorus is a question about which we are struggling since long. When I was a student, we are working without. So maybe I missed it. Did you use neem coated urea also or not?
Sir, actually, we according to new urea policy from 2017, we are whatever urea we are getting, entire urea is coated with neem coat.
So it is neem coated. Okay,
yes, sir,
good.
neem coated.
Okay, good. That's good. Then next one question is, I will say very well planned, but when we go for garlic, etc., if there's some preliminary study, that's better, because once it goes into thesis or into the publication, so we assume that some work or some lead is there already. Only then we'll go for systematic work. That was one point. And second point is quality of publication. When it is only one location, I will not expect better publication in agronomy. That's problem of agronomy and plant breeding. Soil science is still better than us because they do some lab work, etc. For that, my suggestion to colleagues, again, general suggestion, why not you lay another experiment at ISAPUR farm if Dr. Bansal's successor agrees? ISAPUR is just Delhi, and that is altogether different agroecology. In fact, Dr. Ekising at PU, I said made it compulsory. 2 years or 2 location data. So that's outside the scope. Pooja, you should not worry about that. And then another general suggestion again. We overemphasize negative things about agriculture. Food is important. It is must, we must eat. It is not must that we must have a big car. It is not must that we must smoke. I can just go on counting these things. So let us put in fact, again, for information, when I was AD, 2 things happened. One was publication on a disease. Dr. A.K. Singh, I have forgotten, it was wheat disease, some snow white, etc. from PAU, which cannot be there in India. Second thing was publication from Environment Division, Environment Science Division of IR is established. It was about gas emission from puddled rice. And then ICAR issued instructions that whenever new disease or insect is reported, must take permission. And similarly, we should be careful about environmental publication also. Of course, that goes against the science. But national interest comes number 1. So very good, wonderful. Publication are weak, but we cannot expect better than that. And please be balanced. Food is must. We have to eat rice. And to fill our belly, we have to go for puddle, unless we go. Now we are making direct seed rice also. Wonderful, Bete. Very good. And many congratulations for many awards, etc., etc.
Thank you, sir.
Haji, Dr. Duhujasa, please. Let us move it.
Yeah. Dr. Burman, could you receive the slides from Pratiksha?
No, sir, not yet.
Okay, so then I think we can move on to the next presentation, which is by Ms. Adiga Amla from Floriculture and Landscape Architecture.
Respected Chairman Sir, jury members, eminent scientists, and my dear friends, I welcome you all to my presentation. Floriculture is the only profession in the world which gives money for being near to the nature. It includes cut flowers, loose flowers, cut grains, of which loose flowers occupies the largest share, among which marigolds ranks first. Majorly produced in countries like Mexico, Brazil, Western Europe, Iran, Georgia, Cambodia, and naming a few. In India, marigold occupies the top position in loose flower market, taking a big swipe across different states, taking a share of 18% from total area under flowers and 27% from total flower production in the country. Marigold, a versatile crop, majorly used as a loose flower crop, a source of natural color, dyes and poultry feed, well-proven herbal healer, used in rotation with several other crops against nematodes and used as an insect repellent. Farmers grow different marigold varieties in 3 different seasons, but during winter season, especially under northern Indian conditions, the production of marigold is limited due to the lack of cold-tolerant varieties. So the plants sensitive to cold stress, they must be grown under greenhouse or covered under polythene cover for quality flower production, but farmers can't afford the high cost of greenhouse. No report of cold-tolerant varieties, thus comes the importance of development of cold-tolerant marigold phenotypes. Before that, it is imperative to understand the effect of specific stress factors on various physiological, biochemical, and morphological parameters. Some of the morphological changes like blackening of the stem, water circulations on the flowers, and finally wilting and death of the plant. And physiological changes like loss of chlorophyll, accumulation of anthocyanins, and formation of asbestos leads to damage to the cell membrane and leakage of electrolytes and water molecules from the cell membrane. And biochemical changes like the wilted plant shows less accumulation of osmolytes, and the cell with oxidative stress shows less accumulation of antioxidants, thus rising the production of free radicals. This is the normal cell, the cell attacked with free radicals, and the cell with oxidative stress with no antioxidant defense system. In order to avoid this stress, plant undergo a process called cold acclimatization, where the cold-tolerant plant cell shows higher accumulation of osmolytes and high production of antioxidant enzymes with less or worse damage to the membrane, with inactive leakage of electrolytes and high transpiration and high photosynthetic rate. So the cold-tolerant and cold-susceptible varieties shows higher yield and lesser yield respectively. With this background, I, E.D. Gamala, representing from the discipline of Floriculture and Landscape Architecture, worked on cold-tolerant studies in marigold tester species under the guidance of Dr. Kanwapal Singh. with two objectives. The first one is to screen marigold xenotypes for cold tolerance based on morphological, physiological, and biochemical traits. And the second one is to find phenotype of marigold xenotypes influenced by low temperature through high throughput phenotyping. Coming to material and methods, 10 xenotypes of African marigold, Tastus erecta, 10 xenotypes of French marigold, Tastus petrula. These were grown under two different conditions, open and polyhost, with two different sets of experiment. The first set started from mid-November to mid-February, and the second set from December into March to see the effect of cold stress on well-grown and young-grown plants in December and January. The lowest temperature recorded in the month of December and January, where it dropped to almost minus 0.8 degrees Celsius in the first week of January. Under first objective, 3 experiments were conducted. In first experiment, following vegetative and flowering traits were recorded. Coming to results in African marigolds, here the first set represents the marigold xenotypes grown from mid-November to mid-February, and the second set from December into March. The plant head at 75 days after transplanting. This was found higher in the second set with 65.84 centimeters in AFW4 xenotype. Higher number of secondary branches per plant. Higher is the plant spread observed in well-grown marigold xenotypes, where Pusa Bahar ranks first. FW1 shows less number of days for flower bud initiation in polyhost conditions grown from mid-November to mid-February. Breeding for early flowering is an important trait under cold stress conditions. Early grown marigold xenotype shows less number of days for full bloom with only 73 days observed in the xenotype AFW8. Pusa Bahar was found with maximum flower diameter and high flowering with 262.69 gram per plant grown from mid-November to mid-February. In French marigolds, plant head at 75 days after transplanting with 42.40 cm and higher number of secondary branches per plant was observed in the xenotype Pusa arpita. Well-grown marigold xenotype shows maximum plant spread with 39.97 cm in Pusa arpita. The intimidator shows least number of days for flower bud initiation and only 41 days for full bloom observed under polyhouse conditions from mid-November to mid-February. Orange winner was found with maximum flower diameter but high flower yield with 230 gram per plant was observed in the xenotype Xrd beauty grown from mid-November to mid-February. In experiment 2, following physiological traits were recorded. Results in African marigolds, the less leakage of electrolytes and higher photosynthetic rate was observed in the xenotype AFW4 in first set. The first set polyhouse grown marigold xenotype shows higher transpiration rate in the xenotype AFW4. In French marigolds, young plant shows high leakage of electrolytes, Valencia yellow and Xrd beauty with highest and lowest ELR respectively, and highest photosynthetic rate was observed on well-grown polyhouse marigold xenotypes Xrd Jeffrey. Xrd beauty was found with maximum transpiration rate under polyhouse conditions grown from mid-November to mid-February. In experiment 3, following biochemical traits have been recorded. In African marigolds, the total chlorophyll and the total phenolic content, this was found maximum in well-grown marigold xenotypes AFW4. The total soluble sugars and the proline, these are an important osmoprotectants under stress conditions, was produced more in the first set grown marigold xenotypes AFW4 with 393.21 mg and 9.72 micromole per gram respectively. Higher the antioxidant enzyme superoxide dismutase, less is the malonaldehyde content production, which is a byproduct of lipid peroxidation, was found in AFW4. In French marigolds, the total chlorophyll and the total soluble sugars was found more in Xrd Jeffrey xenotype with 12 and 450 mg per gram respectively. Higher the Xrd enzyme production and higher phenolic content was observed more under outside grown marigold xenotypes in the first set in Xrd beauty xenotype. Higher proline and less malonaldehyde content production was observed in well-grown marigold xenotypes in Xrd beauty. To screen large number of xenotypes through physiological and biochemical analysis requires large manpower, so there is a need to screen large number of xenotypes which requires short period of time but with high accuracy. The answer to such question is high throughput phenotyping. These are fully automated facilities with precise environmental control to assess plant growth and performance by using remote sensing techniques. To know how Z/A interaction influencing the phenotype of the plant. And advantages like 10 times more efficient, spot smaller differences, acquire important data, quantify more complex size, data storage technologies, precision in analysis, short time required, and selection of superior xenotypes. Under Objective 2, following image-based traits have been carried out at Nanaji Deshmukh Plant Phenomics Center, IRA, New Delhi. In African marigolds, various image-based traits like plant area, convex hull, and caliper length, this was found maximum under polyhouse grown marigold xenotypes in the first set in AFW4. This parameter was recorded by using visual camera, and this is the image of AFW4 xenotype grown in polyhouse conditions. The highest water content was observed in the xenotype AFW4. This is the near-infrared screened image of AFW4 xenotype showing water content. And in French marigold genotypes, the plant area, the convex hull, this was found maximum under polyhost condition grown marigold xenotypes Xrd Jeffrey. But the water content was found maximum in the xenotype Xrd Beauty grown in the first set, and this is the near-infrared screened image of Xrd Beauty. Highest caliper length was observed in the xenotype Pusa arpita with 0.89 kilopixels in the first set. The results of the first objective and the second objective were similar in terms of the water content, plant area and greenness and so on. Based on the assay of various physiological, biochemical, morphological and image-based traits, the following salient findings of this research are: among all the African marigold genotypes, AFW4 was found to be the most cold tolerant, followed by Pusa Bahar, and in French marigolds, Xrd Beauty followed by Xrd Jeffrey and Pusa arpita were found to be the cold tolerant, and in between the two sets of the experiment, well-grown marigold genotypes were more colder tolerant, and in between the two species, the French marigolds were more colder tolerant than African marigolds. Practical implications of this study are: farmers can grow these cold tolerant marigold genotypes in the field conditions during the extreme winter season, especially under northern Indian conditions, for getting higher yield. Path ahead, although AFW4 was shown to be the more cold tolerant, but in terms of yield, Pusa Bahar was higher, so these two genotypes can be employed in the breeding program as parents to develop high yielding cold tolerant cultivars. The further research into the gene expression profiles of key antioxidant enzymes should be carried out on marigold genotypes. With this research work, I have published one paper with NAS rating 5.23 and one more got accepted in Indian Journal of Agriculture Sciences with NAS rating 6.37. I got the Best Thesis Award and won Best Poster Award and given an oral presentation in an international conference. With this, I would like to thank my chairman, members of Advisory Committee, Head and Professor, Division of FLA, Director IRA, Dean PG School IRA, ICF for JRF, parents and friends. I would like to end my presentation with a quote: "In joy or sadness, flowers are our constant friends." Thank you.
I would invite Dr. Bansal to please start the interaction with the student.
Okay, Ms. Adiga, thank you very much for your presentation. I think it was an exhaustive study, I would say, for a master's student with two objectives and, with so many different genotypes and doing all morphological, physiological, biochemical. And so it was not very clear to me, really speaking, that on what basis, you wanted to select the ones which are most cold tolerant versus most cold susceptible. Anyway, that's my understanding to me. And also, now the question, Ms. Adiga, to you is that which is the critical stage, by the way, which gets affected under field conditions of marigold, to cold stress, which is the most critical stage for the plant growth and development? Are you there, Ms. Adiga?
Adiga, you're on mute.
Unmute yourself, Adiga.
Yes, sir.
Picture on, yes. Which is the which is the most critical stage for cold stress in marigold under field conditions?
Thank you for your question, sir. As for our study, sir, the young plants, these are more susceptible for the cold stress conditions.
What did you say? Which plant?
Young plants at the earlier stages.
Okay, earlier stages, you mean?
Yes.
But also I was wondering, see, you given kind of a standard treatment, for cold stress, and you compared so many genotypes. So were they not different in the duration or in the different stages when they achieve, we call it as phenological stages, were different? Did you take that into account?
Sir, I'm not clear with your question, sir.
My question is that they must be all different genotypes you selected for your study must be with kind of, different stages in their growth and development in terms of duration to next stage and, phenologically. Were they different?
Yes, sir, they were different, sir.
So how did you then manage the treatment, or your data on that particular stage you taken, or what stage did you take your observations of plant growth and development?
So for I have taken like the well-grown plants I have taken in the month of January, so when they are vegetative parameters I have taken when they are fully grown, and flowering traits when they have achieved the fully flowering parameters. At that time I have taken, sir. But for the analysis of biochemical parameters, I have taken the leaf samples when the lowest temperature was recorded in the month of January. That is the major traits, sir. Major
Okay,
I have
so
for consideration, which is cold tolerant, which is cold susceptible.
what is the difference between objective 1 and 2? When you have a high throughput screening methods available, so it's better to screen through high throughput phenotyping, or you should also go to objective 1 then? Why that objective 1 was required?
Sir, the objective 1 is required, sir, to know how much yield we are getting in the terms of morphological, and even in biochemical, sir, we need to know what amount of osmoprotectants under stress conditions they are producing, what amount of antioxidant enzymes, how they are responding different antioxidants under stress conditions.
Okay, thank you.
Thank you, sir.
May I now invite Dr. Joshi, please?
Thank you, Dahuja. Ediga ji, I think you have a unique voice. You have a unique voice. And I think you can run 100 meters very fast, I'm sure. Because covering such a large number of slides, such a short time, very good. And this subject, is poor man's crop, now flower. Farmers are suffering. Topic is really, I like it. Because I have seen farmers suffering. My question is, how many cultivable genotypes or varieties of marigold are available in NBBGR or any other collection? Do you have idea?
Sir, I'm not that much clear, sir. Maybe around 100 genotypes are available.
Around 100. Yeah, okay. This is I wanted to know. Thank you. Because, tulip, the national flower of Netherlands, they have collection of, they used to have collection of 4,000 tulip lines in Netherlands. So if there are more, your study will help that study in future, because this is a very important subject. Thank you very much.
Yes.
Thank
you, sir.
Dr. Patra, please.
Yeah, it was excellent presentation, Ms. Ediga. Lot of importance in the national economy when we talk about the flowers. In this, the cold tolerance you have identified, is there any relationship on the flower size, color, yield, and keeping quality of the flowers?
Yes, sir.
What you have found?
So there is a difference between the flower size and flower quality from polyhouse condition and even from open condition. We have observed the flower size have been reduced in the outside conditions compared to the flowers which plants grown in polyhouse conditions. The quality is differing, sir.
Okay, and the color? The color?
So color, sir, like just wilting of the flowers, sir. It is mostly like due to the when temperature is like below 0 degrees Celsius. At that time we have observed the flowers blackening of the flowers. It is almost like killing the tissues in the flowers. In the outside, it's not. It is very quality of the flower is good in the polyhouse conditions.
Okay, thank you. All the best.
Thank you, sir.
Dr. Patil, please.
Miss Ediga, during COVID-19, actually this floriculture is the worst affected actually area because our temples were closed, there were no shadis and all these things, and you have selected this topic during this pandemic of COVID-19. So which force you have drive to select this topic for your MSc?
Sir, actually, thank you for your question, sir. This is nice question. And marigold, sir, this is a major loose flower crop which is grown in south side conditions and even northern side, sir. And no work has been done, sir. Actually, no research up to now. I didn't find any literature cited on this work. So me along with my guide, sir, I'm thankful to my guide to select this new topic for working on this. So we have to go further in this research work, sir.
Thank you. Thank you.
Thank you, sir.
May we now have the observations of the chairperson?
Yeah, thank you, Dr. Saab. Thank you very much. And congratulations to Ms. Amala for good presentation. And I will say very exhaustive study for MSc student. Working out so many things. So I don't have any question because my colleague have raised the pertinent question, but I would like to share my experience with winter maize. First of all, so it's maybe a little bit useful to the guide or other faculty members working on this aspect. First thing is that we have to standardize the stage, phenological stage. For that, you need sowing at different time, then exposure also different time. For that, what Dr. Hansel started with that. Of course, stage will vary from crop to crop. Then for quick screening, it will be, maybe it becomes easier after 5, 10, 20 years, or maybe at IRE, now itself, the facility can be used. But otherwise, we need to have some morphological traits. What we observed in winter maize when we started in 1978, that germplasm from Germany was wonderful for cold tolerance, very poor for yield. That is what you have observed. French genotypes, they are good for cold tolerance. 3 types of symptoms we observed: yellowing, drying of leaves, and purpling. Ultimately, then purpling came out to be the positive indicator. When we saw just in nature, first thing we saw, just guava leaves, I noticed that in winter they turn purple. So that is what, there are some chemical, and that acts. Then we found a publication in crop science also, in not in crop science, in plant breeding also. So these are 3 parameters. Then water management, Dr. Vansal, you raised this issue earlier also. Water management is very, very important. If we have good water management, crop is saved. If we have little bit over irrigation and there is low temperature, particularly frost, crop just gets oily and it just looks burnt crop. So these are the factors which, if Amala, you have to do PhD work on this, you want to continue, or some other want to continue. Ultimately, I think for I will not say unimportant crops, but some crops which are economically not that important as others, you will have to go for morphological traits. For MSc thesis, PhD thesis is wonderful. You have to bring in these things to have good publication. Wonderful, very good. Thank you.
Thank you, sir. So with this, now we'll move to the next presentation, which is by Ms. Rutuparna Senapati from discipline of fruit science.
Respected Chairman, jury members, scientists, seniors, juniors, and my dear friends, a very good afternoon to everyone. Fruits and vegetables are called protective food. FAO declared the year of 2021 as International Year of Fruits and Vegetables. Studies indicate that human ancestors were fruit eaters. Fruit provide a wide range of health-promoting compounds like vitamins, minerals, including antioxidants, and this antioxidant property of fruits is mainly due to pigments. Plant pigments are naturally colored substances and are mainly classified into 4 main categories. They are chlorophyll, carotenoid, flavonoid, and betalains. They directly and indirectly play important role in many physiological processes in plants. These pigments represent specific color in different fruits. Guava, which is called as miracle fruit, it is the 3rd richest source of vitamin C after Barbados cherry and amla. It prevents from many deadly diseases and improves immunity. Guava also a preferred choice of growers because of its wider climatic adaptability, suitability for fresh as well as processing purposes, and profitability. It contributes 4.2% of total area and 4.3% of total fruit production in India. When we look into the variations of pulp color of guava, as per the oldest classification of pulp color by Ruhel, guava can be classified into 4 categories: white, yellow, pink and purple pulp color. However, the recent classification as per UPOB did not mention the purple pulp color. The white pulp guava are rich source of vitamin C, yellow color guava contain more amount of lutein, pink pulp guava is known for their higher lycopene content, but the pigment responsible for the distinct pulp color of black guava is not known. My senior Hanuman sir in 2020 reported that the lycopene content in black guava was found very negligible. So he concluded that the purple pulp color may be due to the accumulation of some other pigments which may be synthesized independent of lycopene biosynthesis pathway. So the research gap which is identified was which pigment is responsible for the pulp color in black guava. With this background, I am Rutupana Senapati from the Division of Fruits and Horticultural Technology, taking you all into my research topic on understanding the basis of pulp color in black guava. In our study, the objective one is to estimate the metabolites responsible for pulp color in black guava, and objective 2 is to characterize the gene involved in metabolite synthesis in black guava. In plant materials black guava, Risar Surka, Punjabing, Lalit, Ponprabhat, Alava Saveda, Trichi and Sweta fruits are taken. And in objective 1, morphological, physical and biochemical characterization of the fruits were done, and the data are subjected to statistical analysis, where ANOVA and Tukey's honest significant difference test was used to identify the significant difference and pairwise significant difference among the genotypes. The morphological characterization clearly indicates the distinctness of black guava in tree as well as leaf character in comparison to other 7 guava genotypes. Then moving to another morphological character, that is fruit character, the peel and pulp color both are the economic traits and were observed by the Royal Horticultural Society color chart, and it was found that the peel and pulp color of black guava belonged to the grade orange group resembling purplish in color, which was entirely different from other 7 guava genotypes. The physical parameters like fruit weight, diameter, length, seed coat, diameter and pulp thickness were observed by the standard methods. The data on physical fruit parameters indicates that for most of the physical fruit characters, the black guava belongs to the intermediate category. Under biochemical parameter, maximum TSS was recorded in Hisar Surka, which differed non-significantly with black guava, and the black guava also had maximum total sugar content. Total carotenoid content was measured by the protocol proposed by Sadler and his co-workers in 1990. The results pertaining to the total carotenoid in 8 guava genotypes is very interesting, and irrespective of the pulp color, that is pink or white, total carotenoid was present in all the 8 guava genotypes, and black guava recorded maximum amount of total carotenoid in it. Total lycopene content was measured by the protocol given by Godwin with his co-workers in 2015. The results of lycopene content estimation convey that among the 8 guava genotypes, 4 were colored and 4 were white pulp. Black guava, which also belonged to the colored guava group, contained little amount of lycopene as compared to the remaining colored guava genotypes. Maximum lycopene content was recorded in Hisar Surka, and all the 4 white pulp guava genotypes had negligible amount of lycopene in them. Total anthocyanin content was measured by the protocol given by Glusti and his co-workers in 2001. It is very interesting to know that maximum total anthocyanin content was found in black guava, which belonged to the distinct color class red orange group. It was followed by Hisar Surka, a pink pulp guava genotype. Pon Prabhat is a white pulp guava genotype, and the presence of anthocyanin in it may be due to the pigmentation on the fruit peel due to the low temperature, and remaining white pulp guava genotype and negligible amount of anthocyanin in them. The comparative graph of these pigments clearly indicates that black guava had more total anthocyanin content, that is 9.663 mg per 100 gram, as compared to lycopene and total carotenoid. Whereas pink pulp guava genotypes contain lycopene and anthocyanin both or lycopene alone, and total carotenoid were present irrespective of the pulp color. As the analysis showed that black guava had more total anthocyanin content, so LCMS was done to find out the tentative compounds. This is the extraction protocol. The data of LCMS indicates that based on the mass and charge value and deuterion, 3 compounds were identified in black guava. They are delphinidine 3-glucoside, petronidine 3-glucoside and cyanidine 3-glucoside, which are responsible for its unique pulp color. To confirm the LCMS results, HPLC was done. In this, the presence of these 3 anthocyanin compounds were confirmed in black guava, but they were absent in white pulp Pon Prabhat. The salient findings from the objective 1 are maximum total anthocyanin content found in black guava, and it is minimum in Punjab pink. Maximum lycopene content found in Hisar Surka and minimum in Pon Prabhat. From the LCMS and HPLC, it was found that 3 anthocyanin like delphinidine 3-glucoside, petronidine 3-glucoside, and cyanidine 3-glucoside are responsible for the unique pulp color in black guava. And the very interesting fact is found that irrespective of carotenoid found in all the 8 guava genotypes. Then moving into our second objective, characterization of the gene involved in metabolite synthesis in black guava. As anthocyanins were responsible pigments for pulp color of black guava, so anthocyanin pathway genes CHS and DFR were selected for objective 2. Before going to the details of the study, I would like to say that why we choose this gene for the study. The reasons are CHS, which is required for the formation of naringenin chalcone, is the fast committed step which leads to the synthesis of anthocyanins, and the dihydroplavonol reductase, which forms the leucoanthocyanidin, is very closest process to final anthocyanin formation. As these two genes are very close to the first and last step of anthocyanin biosynthesis, so we include them for molecular characterization. Genomic DNA was extracted from the black guava and Punjab pink, as black guava contained maximum total anthocyanin and Punjab pink contained minimum total anthocyanins. Then in designing of primers, 11 primers were designed, 5 primers for CHS gene, 6 primers for DFR gene. This band statistic shows that 3 CHS primers C1a, C1 and C3 out of 5 CHS primers and 2 DFR primers D4 and D5 out of 6 DFR primers were amplified in Punjab pink but not in black guava. The salient findings from the second objective are the primers of DFR and CHS gene were amplified in Punjab pink but not in black guava. This may be due to the fact that the genes responsible for anthocyanins in black guava is structurally different from the other guava genotypes. And second reason could be that both the gene belongs to the multigene family. In conclusion, I would like to say that the distinct purplish pulp color in black guava is due to the presence of delphinidine 3-glucoside, petronidine 3-glucoside and cyanidine 3-glucoside. Pink pulp guava genotypes contain both anthocyanin and lycopene. Irrespective of the pulp color in guava, total carotenoids were present in all the guava genotypes. Thus, it can be concluded that the pulp and peel color in guava is due to the combination of more than one pigment, and the final color depends on the relative proportion of those pigments. So, the statement for the guava pulp color, pigments in proportion, is correct. The future studies will be directed towards the development of comprehensive genomic resources for black guava, especially for peel and pulp color, development and analysis of mapping population involving black guava and low or no anthocyanin varieties, and combining genetic and molecular analysis of the anthocyanin pathway genes in guava. The nobility of our work lies in the fact that this was the first approach on the study of unique pulp color of black guava. With this research, I have published one research paper in Annals of Plant and Soil Research and another paper is submitted in Indian Journal of Agricultural Sciences and is under review. And I got accession number of 3 nucleotide sequences. I have attended several national and international conferences and also rewarded with Best Poster Award in the International Conference on Biotechnological Initiative for Climate Resilient Agriculture. Now I would like to acknowledge my Chairman, Advisory Committee and everyone who supported me during the course of my post-graduation. Finally, I would like to conclude my presentation with a beautiful quote, "Fill your plate with the colors of the rainbow. What please the eye, please the body." Thank you. Thank you very much.
May I invite Dr. Bansal to please interact with the student?
Okay, thank you. What was your name? Miss
Rutuparna.
Thank you. Yeah, Miss Senapati. Right. Well, anyway, I think the objective was clear. But however, I didn't realize, sometimes I know you need little data to fill up your thesis, that what was the necessity when it was so focused, objective, or even your title. Why did you then venture into taking all those morphological data, on fruit size, and et cetera? I know it won't take probably much time, but it has nothing to do with, otherwise, I believe the objective you're trying to achieve, as it was clear from your title as well. So that was one thing, that why these morphology and physical food parameters were identified, and, time was spent on it. You would have certainly gone directly into the deep, biochemical analysis, which you have tried to, and could finally conclude that what could be the reason, and what's contributing, particularly 3 types of anthocyanins. You mentioned it is fine. So can you tell me a little bit about it so far? Then I'll ask you the second question after this, that what was the necessity of going into these food parameters? If you have any specific answer, I thought.
Good afternoon, sir, and thank you for your question, sir. Sir, we take the morphological parameters to know the variations among the other guava genotypes with the black guava. because it is morphologically also different from the other guava genotypes. In plant characters, like tree characters, the branches are erect and also young shoots are red in color, sir. So it should be a morphological marker for the F1 progeny. And then in leaf character, the leaves are very densely pubescent, and in fruit character.
Okay, anyway, okay, fine, thank you. I don't want to waste time there because I have another question. And anyway, certainly there will be always differences, when you measure, compare different genotypes, whether different color or no different color, they will always be different anyway, morphologically speaking, because that's why they're different. And second question is that you went into your identification of the gene, and then you found that probably did not give you enough data or enough information that which particular gene must be responsible for higher anthocyanin in black guava. And you said you design primers. So did you design primers based on some conserved sequence of which particular gene?
Yes, sir.
How did you design your primers?
Sir, first we blast this guava reference genome with the anthocyanin-rich fruits like strawberry, apple, etc. And this is matched, very much closely matched with the apple genes. And then the genes, the guava genome data were also taken from the NCVI, and then the genes were extracted by the use of weight tool in the Linux windows, and overlapping primer were designed.
So finally, what is contributing to these more anthocyanins? Is the pulp or the peel? Do you find any difference between the biochemical constituents in the pulp versus peel in this particular black guava?
Sir, I did not find the difference between the peel and amount of anthocyanin in peel and pulp, but it is more in peel, I think, sir.
Okay, thank you. All the best.
Thank you, sir.
Dr. Joshi, please.
Thank you. Rutuparna ji, I like your statement. Color is very important. But a Senapati should like, that orange color. So can you tell me what is so special about guava and when it is of that color which you are trying to investigate?
It is different from color-wise. It is purple pulp color, and it is very unique. And the unique pulp color is due to anthocyanin. And as it was already reported that the anthocyanin-rich fruits were having many more health benefits, like they prevent from many deadly diseases. So this variety will be helpful in developing nutritious varieties. Sir, also it is having hard pulp and crispiness is in it. So due to this, it is having long shelf life. So this character should be incorporated. If we take the black guava as a parent, we can incorporate this character into the progeny, sir.
Thank you. Thank you. I like guava very much, just like Dr. Dhillon. So thank you very much.
Thank you.
So if there are no more raised hands, so then may I request the chairperson for the remarks?
Okay, Dr. Sir. Okay. I like guava, Dr. Joshi. That is the fruit which has maximum potential in Punjab now. And nutritionally, I was really surprised to know that it is much better, not better, much, much better. It's not good English, but this is what I want to convey. It's much, much better than apple. And we started pushing it, and 1 year full on all melas, our theme was nutritional quality of guava. I am very happy that Ms. Senapati worked on guava, wonderful. And breeding is also very easy. It is not like mango or other crops. It is so fast among all. So then general comment about what I like and what are difficulty. Again, for difficulty colleagues, shelf life is important if you can work on that in this case. Miss Senapati, all these colors, they are controlled by major genes. Do we know number of genes in this case? In fact, there must be different genes. We have apple-colored peel and white pulp. We have pink pulp and white peel, and
Yes, sir.
then black guava also. So there must be different genes, or I expect it, not must, but I expect different genes. Do we have that information?
Sir, no, sir.
That should be the first thing. But okay, for good publication again, yeah, that is the dichotomy. For good publication, we go to primers and those things, etc., etc. Anyway, thank you very much. Very good publication. Very good work for MSc student. Wonderful.
Thank you.
Thank you.
Thank you, sir. So in the sequence, the next presentation is from Mr. Das Gupta from Discipline of Genetics and Plant Breeding.
A very good morning to the chairman and designated members of the presentation committee, scientists, juniors, seniors, and my batchmates. I am Uttaran Dasgupta from Division of Genetics, and welcome to my merit medal presentation. Mung bean is the third most important pulse of India in terms of production and consumption, and India alone accounts for 30% of the global production. Despite having high production, the productivity is low due to various reasons, including abiotic and biotic stresses. Yellow mosaic disease is the most economically damaging disease of mung bean, and the loss can range anywhere from 37% to 84%, depending upon the genotype and the growth stage infected. It is also responsible for the outbreak in 1977 in northern Thailand and also in 2008 in Indonesia. More than $300 million loss has occurred due to this YMD. It is caused by bigomoviruses, out of which the main two are MYMV or mung bean yellow mosaic virus, which is more prevalent in the south and western conditions, and MYMIV is more prevalent in the northern, central, and eastern India. For our study, we had taken this MYMIV due to its prevalence in Delhi. Bigomoviruses have bipartite single-stranded DNA genome and infects a multitude of pulses ranging from cowpea to soybean. It is spread through polyphagous pest whitefly in circulative manner. One of the major control measures in field is by applying insecticides to stop the vectors and restrain the viral dispersion, which is not an eco-friendly approach. Instead, we can go for developing resistant varieties, which is an eco-friendly approach. Now, there are certain limitations in resistance breeding. The first one is that there is limited knowledge regarding the genetic and molecular basis of viral diseases, and also lack of sufficient molecular markers for screening and marker-assisted breeding purpose. In this backdrop, I would like to mention the title of my thesis, which is Comparative Ionistic Analysis and Antioxidant Studies to understand the complex regulatory network of MYMI resistance in mung bean. We have formulated the following three objectives, out of which the first one was identification of key genes and signaling pathways involved in imparting defense against YMD in mung bean. My second objective was identification and validation of novel AST SRs in a selected set of mung bean genotypes differing for MYMIV resistance. And my third objective was investigating the role of phenolics and flavonoids in YMD expression in a selected set of mung bean genotypes. These are the 32 genotypes taken for my study from diverse genetic backgrounds. This is the disease rating scale used, and by using the infector road technique, we had identified 5 resistant, 20 moderately resistant, 2 moderately susceptible, and 5 susceptible genotypes, out of which we had taken the contrasting resistant PMR1 and susceptible Pusa visual for our RNA sequencing studies. For our first objective, we conducted an ionistic analysis. First, we sowed the seeds in glasshouse and artificial inoculation was done using force feeding by villifers whiteflies. We had taken the samples in 4 combinations, that is resistant controlled, susceptible controlled, resistant inoculated, and susceptible inoculated. And confirmation of MIYV was done in the inoculated sample using coat protein specific primer. For our RNA-seq study, 7-day-old apical leaves were taken and 3 biological replications pooled. We subsequently isolated the RNA and sequencing was done using Illumina HiSeq. As we have reference genome and transcriptome, we went ahead with the following RNA-seq data analysis pipeline. We got around 73 million clean reads, and for each sample it ranged from 16 to 20 million, which is enough for RNA sequencing considering the size of the mung bean, which is comparatively small. Our overall alignment rate was also high due to providing greater depth of sequencing and using latest sequence genome for alignment. Our main aim was to find up and down regulated genes, and we identified them in different log fold changes, but considered only the log to fold change as threshold. The most number of transcripts was found in the resistant inoculate and resistant control combination, and also the most uniquely expressing transcripts was found in this combination, signifying that differences in disease reaction is more due to activation of genes in the resistant genotype. We generated this heat map using a set of stress-specific DEGs, which includes genes like LOX-JAZ, which are important signaling molecules in the jasmonic acid pathway, transcription factor family WORDKey, which is related to MAP kinase cascade leading to disease resistance, and NAC transcription factor, which has been reported to interact with the replication initiator protein of MYMIV, leading to reduction in infection. This too has also been found to be upregulated in the RNA-seq study conducted in black gram against MYMIV. MYV is a large family of transcription factor which gets modulated by this jasmonic acid pathway and is responsible for activating the expression of PR genes, which includes the PR protein grade6 family, PR protein 5, leading to resistance. To understand the molecular pathways involved, there is a need to do functional enrichment studies, which is the gene ontology-based classification of DEGs, and also plotted GO terms in scatter plot. Enriched terms included response to jasmonic acid and ethylene, which indicated role of jasmonic acid ethylene mediated defense response against the viral infection. To envision the biochemical pathways involved, we mapped the DEGs into the KEK pathway database and got enriched pathways, photosynthesis and carotenoid biosynthesis, genes of which are target of MYMIV for producing the characteristic symptom. Protein-protein interaction network was constructed by using our RNA sequencing data, and I want to mention that this is the first time which has done in mung bean, and in the resistant genotype we identified 10 interacting protein modules, while in susceptible genotype we've identified 5 interacting protein modules. Integrating all the data, we constructed the complex network underlying YMV infection in mung bean. It included upregulation of kinases, upregulation of JAZ-LOX protein, which leads to jasmonic acid similar defense response, and upregulation of the various transcriptor factor families and expression of PR proteins, all of this leading to the resistance. While the suppression of chloroplast mediated defense and inhibition of photosynthesis genes leads to susceptibility and also production of the characteristic symptom. I want to enlighten that this is the first study to decipher the pathway underlying MYMIV resistance in mung bean. Now if I have identified all these genes, how to integrate them in my breeding purpose? Now in the picture comes my second objective, in where using our RNA-seq data, online microsatellite and batch primer 3 tool, we detected and designed 513 novel stress-specific ESTSRs covering all the 11 chromosomes, out of which 35 was used in our present study. In a recent study by Mathivatana et al., they got a QTL on chromosome 4 harboring 18 candidate disease-resistant genes, so we put more emphasis on designing markers from this region. We ran 76 markers on 32 genotypes, which also included other previously reported markers for which validation studies have not been done yet. Our major findings include 12 polymorphic markers, out of which 4 were novel, but they were unable to differentiate between resistant and susceptible genotypes, and the 10 primer pairs designed from chromosome 4 were also found to be monomorphic, so there is a need to search for more primers to find the MYMIV specific marker. Now coming to our objective 3, from our RNA sequencing data, we got upregulation of certain genes associated with biosynthesis of phenolic compounds, and in a study by Seth et al., they showed that total phenolic content was positively correlated with yellow vein mosaic virus resistance in okra. So we went ahead with the antioxidant analysis. We took 4 stages, out of which the sprout and seedling stage, they were divided in infection, and these 2 stages which were taken from field were under disease prevailing conditions. We estimated TPC and TFC and conducted appropriate statistical analysis. In case of total phenolic content, we got that when there was no disease, irrespective of the genotype being resistant or susceptible, they showed comparable amount of TPC. But in presence of viral infection, there was an increased trend of TPC in the resistant genotypes. Our study clearly indicated that there is correlation between TPC and MIV resistant susceptibility in mung bean. But in case of TFC, what we got that there was no difference in resistant susceptible genotypes in the content, irrespective of presence or absence of viral infection. So TFC was found to be unrelated to MYMIV's resistant susceptibility in mung bean. Coming to the conclusion of our study, 5 genotypes were identified each as resistant and susceptible, and the molecular regulatory network underlying MYMIV resistance was deciphered using the Ionistic Analysis, and a set of 530 novel stress-specific ESTSR were designed, and TPC was found to be correlated while TFC was found uncorrelated with resistance to MYMIV infection. Our study unfolded the pathways and genes involved which can be targeted further for deeper understanding of the resistance mechanism, and also design primers can also be used for finding linked marker for other stresses, including this YMD also. And the correlation identified between TPC with that of YMD can be used as a parameter for screening and breeding purposes. In future, key genes like MYB and Worthy transcription factors LOX and JAS can be further targeted to be used in our breeding programs, and there is a need to find ESTSR markers linked with MYB resistance for marker-assisted breeding. And also there is a need to find the correlation between MYB resistance with some other biochemical parameters and also in other pulses. I published a paper in PLOS One which has an R rating of 9.24 and has also received citations. I have also published a review paper in Frontiers in Plant Science. I've also published book chapters and submitted 4 sequences to NCBI in Sequence Archive. I participated in various national and international conferences. I have qualified ICR NET and GATE license 2021. I have also attended some workshops and I am a member of Genetics. Finally, I want to acknowledge my chairman, members of advisory committee, head and professor, all my teachers, and our beloved teacher, Director Sir IRI. Finally, I want to end my presentation with the quote of eminent scientist Dr. M.S. Swaminathan that future belongs to the nation with grains, not guns. Thank you.
I would like to invite Dr. Joshi to interact with the student first.
Yes, thank you very much. Good presentation and good publications. The name is very good. You become very important after Makar Sakranti. Uttarayan, very good. So I thought I should ask a question. I want to ask beyond what you have done. So Uttarayanji, do you see, although it's a message, I'm sorry, don't take it, that I am asking a big question. Do you see that the information you are generating or your team is generating would be tomorrow used for gene editing? Because now the days are changing. It is all, genomic selection is also now a smaller term. So gene editing, do you see anything, any linkage in future?
Thank you for your question, sir. Sir, I want to say that the gene editing you have mentioned, sir, we have also found certain genes in the susceptible plant, which are responsible for helping to produce the disease side of the virus.
Thank you. Good. Thank you. Your connection is little weak, but I got your answer. Thank you.
Dr. Bansal, please.
Well, Mr. Dasgupta, I just wanted to ask, see, you have gone into this molecular analysis also and some biochemical analysis, and finally you say something TPC, TFC. I don't know what these are. You should not be using these abbreviations anyway. In fact, really speaking, while making a presentation, unless they are DNA and RNA. And so anyway, TPC is total phenolic content. And you showed a very good relation, between the two, susceptible versus resistant. And, you could find that there's a good correlation, right? Hello?
Yes, sir.
Is that right? What I'm saying is
Am I audible? Yes, sir. Am I audible, sir?
I'm not able to hear you. Please, can you? Hello?
Sir,
Can you hear me?
Am I audible, sir? Yes, sir. I can hear you, sir. Am I audible, sir?
You are audible, but voice is not clear.
Yes. Now tell me, did you hear what is internet connection problem?
Actually, I was out for a period of time, sir. I couldn't hear the questions, sir. Sorry for the inconvenience on my side. I'm really sorry, sir. I'm keeping logged out.
Hello?
Please, am I audible? Sir, am I clear?
Yes, yes. Go ahead, please. Go ahead. Please go ahead.
Yes, sir. Yes, sir. Your question.
My question is, could you hear me what I asked you before? No.
No, sir, he couldn't listen you, sir.
Okay, and then my question was that you ventured into molecular analysis as well as some biochemical parameters you have estimated, and you found that total phenolic content is related with susceptibility versus resistance, and you find this TPC, total phenolic content, more in resistance, right? Is that correct?
Yes, sir.
So, and then you say you would like to have more biochemical estimations also, done, etc. So, do you think there's something
Yes,
more
sir.
which you have reason to say this, or what?
Actually, in the other literature, when Okra by certain cohorts, they thought that even other biochemical parameters like certain ascorbic acid total chlorophyll content, they are also linked to the nutrients. So that's why I mentioned sir other biochemical factors, and in my study also we got certain like the Jasminic acid pathway. So we can use this.
Is it my internet problem or your internet connection problem, Iron?
No, sir, his internet connectivity is poor, sir.
Yeah, looks like, anyway, I think.
Uttarayan, you can text your answer in the chat box.
Yes,
Dr.
I think
Bansal
that's
will see.
better. That's better. And second, if you can hear me, also text me. You also, if you can hear me, did you find any of your gene function when you were doing it, this analysis related with total phenolic content? Anything, any signals you were getting, regarding that?
Am I audible, sir?
You can answer in the chat box to save time. That's better because your connection is a little poor to me, it appears. I don't know whether maybe mine too. So thank you, Mr.
So I'm also.
No, you are not clearly audible. Sorry, but never mind. Go to the chat box, please, and answer your question, and answer it there.
Yes, yes, yes.
There are no more raised hands, sir. So can we have the comments from the chairperson, sir?
Yeah, I don't have any specific comment. Dr. Bansal asked what I wanted. But one thing is there that again, I am very happy with the choice of research projects for MSc student. And just with the MSc work, so good publication. Congratulations to institute, to the student and guide to everyone. Yeah, just for my knowledge. So far I was hearing MYMV. Now it is MIV. Maybe someone else if can explain. Number two, sowing date is an important factor. Are we taking that also into account when we are studying such things? And which is the most resistant gene? I can understand that MS or something like that. Is that from IRA or maybe? Maybe you just write in the chat. It is you are not clear. You are not I mean it's not clear. Speech is not clear. Not clearly audible. So you can write in the chat box.
Sir, now it's audible.
Yes, sometimes yes. But my questions are very simple. The questions are not to you. It's just general knowledge. Maybe your advisor can send a message to me. So I think we can move on further. Very good publications. And one of the important crop which can help in diversification. Wonderful.
Okay, thank you, sir.
Please.
So we move on to the next presentation in the sequence, which is by Ms. Sonam from Discipline of Microbiology.
Honorable Chairman of the session, respected jury members, distinguished scientists, and my dear friends, a very warm welcome to you all. As pointed out by Barack Obama, I firmly believe that no challenge poses a great change to future generation than climate change. As a developing nation, India is confronted with the enormous challenge of supplying accessible energy to increase its economic growth. Meanwhile, striving to meet this energy demand, now India has become world's fourth biggest emitter of carbon dioxide. Therefore, in this grim scenario, questions arise about the climate change are too many. As an answer to this, India is now taking different initiatives to combat the problems of climate change. Newspapers. Even in our budget, they talked a lot about the climate change as well as the renewable energy issues. As you can see here, out of the total energy consumption in the world, only 19% is being built up by the renewable sources. Considering the renewable sources, around 50% is being taken from the biomass. And about the ethanol production, a large portion of the feedstock is contributed by sugarcane and corn, forming the first generation biofuel. However, a small portion of the contributed by crop residue and other sources, forming the second generation biofuel. India, being an agricultural important nation, has more than 90 million tons of crop residue is wasted every year. question raises, can't we utilize these crop residues economically? If yes, what can we do with it? Talking about the advantages of these crop residues, these are low cost, abundant, sustainability, eco-friendly, and obviously being carbon neutral make them a good source for making bioenergy. But again, there are some constraints as well. Being decalcitrant, they require a high energy efficient technology for their degradation. Here, our main villain comes, that is lignin. Already we know that for lignin degradation, many methods are being used, which are physical, chemical, and biological. Among them, the biological conversion method added some advantages like being specific, energy efficient, and less initial investment. But with every positive step, there comes few cons. In case of biological methods, these are less efficient hydrolysis and prolonged time requirement, which brings us to a dilemmatic situation about what to do and what not to. Now, in this context, we can introduce our heroes, that are microorganisms that degrade in biomass in their natural niches. Without any additional input, we can easily get them and can use them for biomass degradation, which becomes a highly efficient system for sustainability of our environment. Generally, different microbes like fungus, bacteria, and actinobacteria are known efficiently to degrade the biomass. Among them, my organism of interest is actinobacteria, the marvelous wonder. These microbes are highly abundant in nature, known to survive in harsh environmental conditions, having excellent lignin depolymerization activity and also continuous enzyme secretion capacity. Since ages, actinobacteria have been used for different purposes like antibiotic, prebiotic, weedicide, and larvicides. After discussing all, here my story comes to a halt now. So far we have discussed, we have excessive crop residue that have to be managed, which brings the next question that we need to identify potential strains for biomass conversion. With this background, I conducted my research work entitled Demonstrating the Lignocellulitic Potential of Actinobacteria for Application in Biomass Processing. Two major objectives, that is, the very first one was to isolate, screen, and select potent actinobacterial strains having lignocellulitic enzymes and to characterize their enzymes. The next one is to evaluate their biomass conversion potential on dry straw. Under my first objective, I isolated actinobacterial strains from compost using enrichment culture technique and did a morphological characterization using gram staining and microscopy. After that, I did a qualitative assessment for lignocellulitic enzymes such as cellulase, gyelanase, lackage, and lignin peroxidase. Based on the enzymatic activities, strain 1, that is isolate 3, and strain 2, that is isolate 7, were found to be potential degraders. Using 16 sRNA sequencing, strain 1 and strain 2 were identified as Streptomyces theophilus and Streptomyces thermophilus respectively. Now in the next step, I did a quantitative assessment of this lignocellulitic activities of the selected strains in submerged fermented state with 1% cellulose or ryestraw as a sole carbon source. The three major observations obtained here are: the ryestraw is a better substrate than cellulose. The second one is, except beta-glucosidase, all the enzymatic activity were higher at 21st day of fermentation than 10th day of incubation. And finally, we observed that the strain 2 performs better than the strain 1. Coming to my second objective, to evaluate their potential on rice straw. Under solid state fermentation, rice straw was treated with the selected strains. Thereafter, 21 days of incubation, it was extracted with both buffer and alkali. The buffer extracts were used for assessing the enzymatic activity, while the alkali extracts were used to estimate the degraded lignin. After that, the collected pre-treated solids were demonstrated for its sugar syrup formation. Now, these are the graphs of SSF buffer extracts. We can clearly observe that strain 2 performs better than strain 1, and also more enzymes were produced in solid state fermentation compared to submerged fermentation. After that, the observance of SSF alkali extracts were taken at 205 nanometer to estimate the lignin extracted. Here, lignin recovery was found to be more in strain 2, that is 18.87 mg as compared to strain 1. This again proves that strain 2 is a better degrader than strain 1. Meanwhile, the lignin recovery was found to be 16.21 mg per 5 gram of rice straw for strain 1, which proves it to be a better degrader of lignin. After lignin removal, the percentage cellulose were estimated. From the table, we got two major findings that when buffer was used as extracting agent, the percentage cellulose enrichment was low, that is 37 to 45% for both the strains. But when NaOH is used along with the strains, it coherently increased the percentage cellulose enrichment by 49 to 53% by degrading more amount of lignin. Comparing the alkylist extracts of both the strains, the percentage enrichment was found to be more in strain 2, which also correlates with better lignin degrading capacity taken as 205 nanometer. After getting to this conclusion, a cell visualization was done to compare the inoculated one with raw one. From these pictures, it is clear that more disintegration were there in alkali extracted sample compared to the buffer one. Also, disintegration was more in inoculated as compared to the uninoculated raw one. Here, in Xrd analysis, we can clearly observe that the HIP at 16 degree and 22 degree indicate the higher exposure of amorphous cellulose in microbial inoculated rice straw as compared to the raw. And also in FTIR, the vibrations at 15-16 nanometer indicates lignin removal was more in treated rice straw as compared to the raw rice straw. Thus, summarizing the silent findings of my research work, actinobacteria predominant in the rice straw and yeast cultures may come out as a great solution for crop residue management and helpful in biomass processing. Streptomyces thermobiolicius and Streptomyces thermophilus are found to be potent biodegradable for their exploitation in biorefineries. These strains are also suitable for mining efficient cellulolytic enzymes production. Under future perspective, my research was practically implemented in developing enzyme cocktails for biorefineries, can be a genetic pool for engineering efficient microorganisms, and also can be commercially deployed for biomass processing. I have delivered oral presentation of my research work in 2 national and international conferences and also actively participated at various conference, symposium, workshops as well as different training programs. I am keen of dancing and actively participated at several cultural events too. Further, I have been active member of different organizations. I qualified CSIRNet, ICRNet and also secured an All India rank of 4 in ICR SRF examination. Now, I would like to thank each and everyone who have helped me in completing my research work successfully. Here, last I am concluding my presentation with a famous quote saying, "Nothing is lost, nothing is created in the operation of art as those of nature." Thank you all.
Yes, I would like to invite Dr. Patra for the interaction with the student.
Thank you. Ms. Sonam, excellent presentation, I must say. Lot of basic work you have done on a very important subject of crop residues management, which is a burning issue. In this context, you know that IRI already developed a PUSA decomposer, which is very much becoming popular and widely being used also. So that is one success story is there, and this is a new kind of studies you are doing with actinobacteria, isn't it? And this is now at the, I think at the laboratory level, you have identified, and then you have seen the preliminary efficiency of these different strains of actinobacteria. So you any decomposition of the crop residues in a small scale, have you started on this, and about the efficiency in comparison to what is existing? Have you evaluated that?
Sir, about the enzyme hydrolysis, we are already using in industry fungus like Trichoderma and Aspergillus. If we compare it, its efficiency with them, then we can found that they are almost similar. Like for Aspergillus, it is 50%, but for Actinobacteria, it is 46 to 45 to 46%.
Okay. And about the time, how much time it is taking, when we comparing?
So when we are comparing, the fungus will take too much of time, and also for them the continuous secretion of enzyme is not there. But if we use the Actinobacteria, we can found that we have an advantage of continuous enzyme secretion, and also the time duration is less, so that we can give our saccharification very easily.
So do you expect it to be still more efficient? Your organisms which you are going to use?
Sir, its enzyme are efficient, but if we can do some genetic modification with it by adding some housekeeping genes before it, so that it will function continuously and can give us good production.
Okay, thank you. All the best.
Thank you, sir.
Thank you, Dr. Patra. May we have now comments from Dr. Joshi?
Thank you, Sonam ji.
Thank you.
Very, very interesting subject. But it's very cold, I don't have a turban like Dr. Dhillon. I am feeling cold. So these organisms also feel very cold. So what is the role of temperature here in increasing or decreasing that decomposition, which we know high temperature more, low temperature less? Anything else you want to add here?
In biorefineries, they are working at a temperature of 40 to 45 degrees Celsius, and also in different pH and temperatures. We conducted this research work concentrating on biorefineries. So can we use that in biorefineries? So I put the temperature and pH as 40 degree and 8.
Okay, thank you.
Yes.
Can I have a small question?
Yes, sir. Dr. Aprol, sir.
Yeah, very briefly for me, I just want to understand the aim of research is to hasten decomposition of residues. You want them to decompose early? Is that the aim of research?
No, sir. This research is already done for fungus like different organisms. But if we compare that, when we taken the metagenomic analysis of compost, we found that in literature it is 40% for Actinobacteria. So it was not exploited so much yet. So I tried to exploit that. So I can use that for commercial production in biorefineries.
Okay. It's not early decomposition. You don't want to decompose early the residues.
So not that much early, but we can use that as the alternative of stubble burning and all. It has a good benefit, so that we can recover lignin from it, and also did lignin valorization on it.
What I'm saying is, scientifically, do you want to residues to decompose earlier? And if so, why? Why not delay the decomposition?
Sir, please, I couldn't understand again.
I don't know. I don't know the answer. But is that that's the purpose? Because I hear a lot of this, Delhi government doing it. We want early decomposition. Is the aim of research to hasten decomposition?
No, sir. Mainly this research is based on biorefinery use.
Okay.
not main aim of
I think
decomposition.
we can have a separate, discussion. I am very happy to see Professor Abral, and thank you so much, sir. I am so moved with your interest that you have joined the presentation and you are listening. We will have a discussion on this aspect as to what is the purpose, and of course you are quite well aware about it, but we can have a discussion on Pusa decomposer separately. Her finding is primarily on improving.
I'm not talking about finding. I'm talking about aim of research. What is the purpose ultimately?
Yes, so you can tell the purpose of your research, what you have done.
So for my purpose of research is to isolate the actinobacteria and exploit them in biorefineries. So can we use the
Biorefinery,
ethanol production there?
Yeah.
Okay. Okay. Thank you very much. Good. Good.
Thank you, sir.
Thank you, sir. So may we now have comments from Dr. Patil?
Yeah. It's a good presentation. For decomposition of biomass, you have used the actinobacteria, and you just now told this kind of, actually it is suitable for small scale. But if you take the scale-up trials in biorefineries having a very huge quantity of making, suppose ethanol you just talked about, it is becoming a limitation because of the growing of the microbes and all these things. So how you will see from that point of view, because many times it is not successful in when we take up these scale-up trials, very big production. So how you can see from that angle?
Sir, in biological conversion, the main demerit is that energy, the hydrolysis efficiency is low for enzyme hydrolysis. And if we are doing best in our enzymes, that we can standardize it with different temperature pressures, which required in biorefinery, we can do it. It will be comparable with the chemical and thermal methods also.
Okay.
Thank you, sir.
So if there are no more questions, so may I now invite the chairperson for his comments?
Yeah, thank you, Dr. Dhuja. First of all, welcome, Dr. Brol. So nice of you that you are sparing time and guiding younger generation. You have an always source of inspiration for us, and you continue to do so now also.
Great to be
welcome, sir. So Dr. Ekasing's ideas came to my mind. Parali decomposition is difficult. Let us first, maybe wheat straw may be easier. It is a fact that sizable quantity of wheat straw is also burnt in Punjab. Only that's not getting that much importance because quantity is much less than that of paddy. Number one. Number two, 85 to 90% of wheat and rice is moving out of Punjab state. Should be focused on. Otherwise, soil health will go down. Already it is being affected and it will go down. Now, bioresource by a biorefinery, how much it is, first of all, MSc student problem, excellent. I don't question that. But how much raw material or paddy straw will be used for that? I mean, we have always say product rice. So, but again, I will not question the work done by MSc student. It is decided by all of us concerned. And I really feel very happy with the choice of the problems project being presented here, whether it is a movie or it is present one or earlier it was on guava. Then about sodium hydroxide washing. Dr. Ekasing, I think you are aware that we have one place, one factory in Abor, NASA, then one is coming up Verbio, Nala. They do some sort of washing. I have visited these two, but I don't remember. So, but they are producing CNG. Then again, I will come to the point which I earlier also emphasized. Why we present data in a negative manner? That's problem with Indian scientists. Now, Biba said, Sonam said that we are at number 4 position. We should always mention, in fact, I wanted to say that also when initially statistics figure was there, presentation, growth rate. Growth rate alone doesn't mean anything. Our per capita income is nearly $2,000 and US is 44, 45, something like that. I don't know exact figures. Always it should be in comparison to the base where we stand. Here also, I feel very happy that we are not number 2. When you talk of total China, we should talk of this production of gases per capita. Then a European Union is not a country. You put a European Union at number 3. But what is science? Science is critical nature and observation, then explaining or thinking why it is so. Just taking from some newspaper or some publication, European Union as a country, that doesn't convey anything, and the total production of wheat, we say that we are number 1, number 2. But I never like that. Okay, government should say, can say. So similarly, growth rate also, the colleagues from economics and statistics are also there. Growth rate should always be in relation to the base, where do we stand. In general, very good. And my only question is application part, Dr. Grol, I think you are also exploring that very point. Application part may be limited, but anyway, universities and IRE, etc., then publication come into, and publication come from such studies.
No, I fully agree. Publications are important and must be done, but each student's work must add up to the overall objectives of the institute's work. You see, that cannot be done in isolation. Ultimately, after all, the entire research process must contribute to our overall objectives.
Agreed, sir. Agreed.
So the students have to be also aware of the overall objectives of research.
That's why MSc and MSc, the guide is more important than.
I agree. I agree too, but you know, after all, it's our responsibility also.
Yeah, I agree.
Because ultimately, how do you decide a problem? You decide a problem because you need to take 50 steps to come to the answer. After all, ultimately, research must transform into policy, and not one piece of research. It might be 50 such MSc theses which will be required. So they must add up ultimately. So
I fully agree with you. In fact, in the forenoon, most of my comments were aimed at the faculty rather than the student. MSc student,
but certainly a brilliant student. I can see the brilliance right throughout her work. I have no, but my overall understanding, I thought needs to improve also. Thank you very much.
Thank you, Dr. Most welcome. Most welcome. So, yeah, thank you, Bete.
Okay, with this now we move to the next presentation, which is by Mr. Gagandeep Singh from discipline of nematology.
Honorable Chairman of the session, respected jury members and scientists, and my dear friends, I welcome all in my presentation. Myself Gagandeep Singh from Division of Nematology. Nematodes can be introduced by the statement of father of nematology, Dr. N.A. Cobb, who stated that if all the matters in the universe except nematodes were swept away, our world would still be dimly recognizable by a film of nematodes. A very true statement. Nematodes found in land to ocean, from living freely to parasitizing animals and plants and occupying almost all the available niches. We study various plant parasitic nematodes parasitizing important crops, but there is a special category of beneficial nematodes called Entomopathogenic Nematodes, in short known as Epns, which can kill about 200 insect species belonging to 23 orders. Heteroptytis and Steinernema are two most important Epn genera that are widely used as biocontrol agents for management of insect pests. As current farming scopes are increasing towards organic farming, there is need of new biopesticides for pest management, and Epn will have major role in insect pest management. It has been suggested that the native bioagents perform better against local insect pests as compared to the introduced bioagents. So for successful pest management, there is need to find out more local species of Epns. So far, on global level, more than 100 species of Steinernema and 16 species of Heteropteris have been described. And from India, 12 species of Steinernema and 3 species of Heteropteris have been reported. Life cycle of Epns comprises of different stages. Only third stage juvenile of Epns found freely moving in soil, and traditional methods rely on this stage to find out Epns from the field. Traditionally, Epns are isolated from soil by Galeria baiting method, in which larvae of Galeria melonilla are provided to soil samples for several days, which get killed by Epns if present in the soil samples, and Epns are isolated from these Galeria cadavers. But there are several limitations of this method, such as 5 to 10 Galeria baits are needed for each sample, and the recovery rate is just 4 to 10%. And method is inefficient and wasteful, especially in large-scale Epn surveys where lots of samples need to be processed. It is very slow and laborious, as it takes 4 to 7 days to give results. All Epn species cannot be recovered by Galeria baits, and morphological identification of Epns requires highly experienced taxonomists. On the other hand, molecular techniques available for Epn detection requires expensive and sophisticated laboratory setups and cannot be usable at point of service. To solve these problems, an insect-free, fast and cost-effective technique to detect Epns from soil samples was required. Here we come up with LAMP technique, which stands for Low Mediated Isothermal Amplification. This method has several features, such as it amplifies target DNA on a single temperature, and it comprises of 4 to 6 sets of primers targeting large regions of specific DNA, which makes it most effective tool to diagnose any targeted organism from n number of populations due to its high specificity. Whole reaction requires about 40 to 90 minutes to produce detectable results. It requires less establishment cost, as it can work with basic laboratory equipments such as dry bath, which can be available at field labs. And so far, hundreds of LAMP assays for diagnosis of different pathogens have been developed. Moreover, different methods of result visualizations are available, and these LAMP assays can be further developed into kits which can be usable at field labs. By observing this research gap and features of LAMP assays, my research was formulated with the title of Detection of Entomopathogenic Nematodes by Loop Mediated Isothermal Amplification, with objectives to develop and validate LAMP protocols for detection of Heteropteris and Steinernema species from total soil DNA. For designing of LAMP primers, sequences of ITS regions of Heteropteris species and sequences of 18S regions of Steinernema species were retrieved through NCBI blast search. And these retrieved sequences were then aligned using Mega10 software to find out common regions from specific sequences, and these common regions were then used to generate LAMP primers through NEB LAMP primer designing tool. Finally, following two different sets of LAMP primers were designed for Heteropteris and Steinernema species. LAMP assays designed for Heteropteris was named as HETLAMP, and assay designed for Steinernema was named as STLAMP. Reaction conditions were optimized according to the requirements of LAMP assays. To find out optimum temperature for the reactions, samples were incubated at range of temperatures between 65 to 70 degrees Celsius, followed by termination of reaction at 80 degrees Celsius for 5 minutes. And optimum time required for the assays were finalized by conducting reactions on different time spans ranging from 15 minutes to 90 minutes. Specificity experiments were carried out to check the specificity of designed assays to its targeted organisms. For that, intergeneric tests was conducted, in which DNA of different nematode genera was tested on the LAMP primers. Also, different species of Heteropteris and Steinernema was tested on their respective LAMP primers. Sensitivity experiments were carried out to determine minimum detection limit of LAMP assays. Steps include DNA extraction from Epn's pellet, followed by quantification of DNA to find out initial concentration, and 10-fold serial dilution of isolated DNA, and the diluted DNA was then tested on their respective LAMP primers. Soil DNA experiments were performed to validate the LAMP assays on soil samples and to check the minimum detection limit of assays from soil DNA samples. At first, 250 mg sterilized soil was taken in each tube containing components for soil DNA extraction, and then Epns were inoculated serially in range of 1 to 100 juveniles per soil sample. One tube kept without inoculation of Epns for use as negative control. Then DNA from these soil samples were isolated and used to test on their respective lamp primers. Reaction temperature for het lamp was optimized at 66 degrees Celsius and for steel lamp at 68 degrees Celsius. The results were visualized by running amplified products on 1.8% agarose gel, in which positive samples showed typical ladder-like band pattern and non-treated control showed blank. Reactions results were also visualized by adding 10% cybergreen dye into amplified products, in which positive amplified samples changes its color from orange to green, and the samples with no amplification remains orange in color. Both het lamp and steel lamp assays worked best in 60 minutes of incubation. In intergeneric experiments, DNF-only heterohepatitis species was amplified by het lamp primers and DNF-only Steinonema species was amplified by steel lamp primers. In intrageneric experiments, het lamp was able to detect DNF all tested species of heterohepatitis and steel lamp was able to detect DNF all tested species of Steinonema. In sensitivity experiments, het lamp assay can detect DNF heterohepatitis up to 0.02 picograms and steel lamp assay can detect DNF Steinonema up to 9.6 picogram per microliter. Both the assays were able to detect DNA of a single juvenile in total DNA from 250 mg soil. In conclusion, two lamp assays has been developed for easy and rapid detection of two EPN genera, one for heterohepatitis species which is named as het lamp and one for Steinonema species which is named as steel lamp. These assays are first of its kind to detect EPNs from soil and to overcome problems with traditional methods. Both assays are proved specific to its targeted EPNs. Moreover, designed assays are highly sensitive to their targeted DNA regions. Both the assays can detect DNA of single juvenile in total DNA from 250 mg soil, and whole process takes about 2 to 3 hours from processing of soil samples to result visualization, and assays require basic laboratory equipments, which will save time and cost when used in large area surveys where large number of samples need to be processed, and an effective baiting can be followed for the positive soil samples to make laborious process easy and simple job. In future, these assays can be further developed into kits for point of service or field level detection of EPNs without need of expert methodologists, and more species specific assays should be developed for important EPNs for rapid identification. From my MSc research, we have published a research paper with NAS rating 9.5. I have received first prize in International Poster Presentation Competition. And I have received first prize in different inter-school competitions organized by IARI. At the end, I would like to pay sincere gratitude to my chairperson, Research Advisory Committee, Division of Nematology, Postgraduate School IARI, Junior Research Fellowship, my parents and friends. I thank each and everyone for supporting me directly or indirectly throughout my MSc tenure. Thank you all.
Any questions from the jury members? Yeah, Dr. Joshi.
Thank you. I'm asking because nobody raised hands. So I thought, let me ask something from Gagandeep. So Gagandeep ji.
Yes, sir. Good afternoon, sir.
Very good. Namaste. Good afternoon. You have a new tool.
Yes, sir.
So are you sure it will be used because nematodes are so important?
Yes, sir.
So can you tell me, nematodes are, suppose you have to tell to somebody, nematodes are very important, we should do survey, we should convince people and planners. How you will say in few words?
Sir, there are two categories if we are widely studied, that plant parasitic nematodes and entomopathogenic nematodes, means which are beneficial and which are harmful for the plant. Sir, no doubt, sir, reports has been said that the Tritocystin nematode or Meladigreen nematodes is most widely damaging pest in the India also. It has been reported. And in Epns, sir, it is wide, means in foreign and India, there is need to be
Okay. Okay, Gagandeep, thank you. I will suggest you. You are a young boy, young man. Okay. See, take one picture.
Yes.
A field, maybe rice field, maybe wheat field. It's not uniform, heterogeneous in crop performance. I have seen that if you do soil survey, you will find some nematode creating problem there. So using that picture and that statement, you can easily convince anybody. Nematodes are important, so your tool is important. Thank you very much.
Thank you, sir.
Mr. Gagandeep.
Yes, sir.
Again, excellent presentation, and congratulations for the research and extracurricular activities you have done during your MSc. Very interesting subject, this one also, and you are developing the kits, isn't it, for the EPNS, and that may be useful also. So in this context, when we are talking the beneficial nematodes, have you come across, you must have taken the soil samples from the different places, and any kind of different managements which promoting these populations of these EPNS, that kind of thing in your research, or you have knowledge in your MSc subject?
Sir, sorry, sir. I didn't understand your question, sir. Please,
No,
can you?
I mean to say, the different management practices are there.
Yes, sir.
Agricultural crops, horticultural crops, this and that, different kinds of management practices, land use systems. And your study is focusing on the beneficial nematodes, isn't it? And that is a good thing. That is what we want, isn't it? We want or we don't want. We want.
Yes,
their population, the beneficial nematodes populations is better for the soil health, isn't it?
Yes, sir.
Am I right? Or
yes, sir.
So if it's so, then have you studied what kind of management, whether it is the crops or whether it is the fertilizers or whether it is the manures and other kind of different combinations which promoting such populations of your EPNS?
So organic systems promote, sir.
Have you studied?
No, sir. No, sir, I haven't seen, sir. Means if we are taking into organic farming, sir, like chemicals are they are like bad in that situation. They're bad in that. So if we need to move on to the biological control, so there are the organic systems are from.
That is your perception. That is your based upon your knowledge, isn't it?
Yes, sir.
Not yet
I'm not
studied.
well sure, sir.
Yeah. Okay. Thank you. All the best.
Thank you, sir.
If there are no more questions, so can we have the comments from the chairperson, sir?
Yeah, thank you, Doctor. Dr. Brule, are you still here? Do you want to ask something? Or any other senior colleague, if there is someone, they are storehouse of knowledge, they have experience over years and years and years. No? Presentation. And beside organic, I don't know how much area will come under organic and how our food security will be affected by that. That is a different thing. But protected cultivation, that is really under attack due to nematodes. So that can be one area. Secondly, again, a general question, I will not ask you that question. Why we are not able to make much headway on biocontrol or even agriculture chemical in the morning? There are discussion on agriculture chemical. When I was student, Dr. Devan, Dr. Parmar was 2-3 years senior to me. He was student when I joined. They were doing so much of work, but we could not really, we need to look into various factors, how to create awareness. That is one factor. But coming to your work, can we have one protocol instead of having two protocols genera-wise? How much information do we lose if we, instead of 66 and 68, we use 67 temperature? Just it is difference of 2 degrees. Of course, 2 degrees are quite a lot. Did you try 67 also?
Yes, sir. Of course, we tried, but it is a very sensitive to the amplification temperature, so we could not find a common temperature for the amplification.
So neither 66 nor 67
No,
nor
sir.
68, it didn't
No,
work
sir.
for both the genera.
No, sir.
Any reason for that?
Sir,
So sensitive.
Sir, due to its requirement of the primers and length and requirement of primers, annealing temperature is most crucial in this step, sir.
No, primer must be depending on, I'm not a biotechnologist, must be depending on genetic diversity, and genetic diversity, Bandar and Bandar are equal.
No, sir. It cannot be.
Excuse me for
sir,
putting in Hindi and Punjabi in one sentence. Anyway, leave it. Let us leave it. I'm
sir,
not biotechnologist, so I
genes
don't
were
know.
also different, for that's why we cannot make it, sir.
Anyway, I have not seen DNA excerpt once. Dr. V.L. Chopra showed me. And that's fine. Thank you. Thank you very much.
Thank you, sir.
Thank you, sir. So now we move on to the next presentation in the sequence, that is by Mr. Sundeep Kumar from Discipline of Plant Genetic Resources.
Namaste. I wholeheartedly welcome all the dignitaries to my thesis. I take pride in saying we the agriculturists feed the world. Today's world is facing security is malnutrition. Pulses play a significant role in mitigating this threat. Rightly, they have been called as the poor man's meat. Black gram, an asiatic vigna species, is truly a potential and versatile pulse crop. But black gram is highly susceptible to Callosobruchus maculatus, the larva being the sole damaging stage feeding on the starchy material of the seed. Its occurrence is widespread around the globe. Bruchid damage in pulses like black gram is manifold. The loss in seed weight and protein content has been reported to vary from 45 to 60% in black gram due to bruchid infestation. The lack of detailed bruchid resistance evaluation coupled with narrow genetic base has resulted in scarcity of reliable resistance sources against Callosobruchus maculatus in cultivated black gram. So the need of the hour is to identify novel and natural resistance sources and widen the available genetic base, which will facilitate introgression of resistance genes. There are various approaches to manage or control storage space like bruchids, but host plant resistance is the relative amount of heritable qualities possessed by the plant which influence the ultimate degree of damage caused by the insects. So how to achieve host plant resistance? The answer lies in plant genetic resources, which are the genetic material of actual or potential value for food and agriculture. With this background, I took up my research work on assessment of genetic diversity and Namaste. I wholeheartedly welcome all the dignitaries to my thesis presentation. I will begin with the quote, "Engineers design the world, doctors cure the world, but I take pride in saying we the agriculture."
Is there any problem, Dr. Burman?
Dr. Burman, this audio was interrupted a lot.
Yes, sir. I am also not sure why it is going wrong. I'll try to play once more, sir.
No, then let's go to the next.
But when we
Let's
test,
go to the next one, and we can have these two presentations at the end. This is problem with the quality of the presentation that you have received, because you see nobody is able to hear, so you can't blame it to net connectivity.
no, sir. Yesterday we tested it. At that time, it was fine. We checked it.
This video was fine, sir.
Yeah.
But maybe because we have, whether the number of participants for 577, that puts a little load, but this is meant for 1000 capacity.
Yes, sir.
So that should not be a problem.
So I'll try once more, sir.
Okay.
But I'm really happy with the number of participants. Really wonderful. I'm noting since morning.
Audio is not there. Dr. Burman, audio.
Yeah, voice is not there.
Voice is not coming, sir?
No.
No, voice is not coming.
Now slides are okay, but voice is not there.
But I can hear, sir. In my system, I have all the sound. So can we?
Sundeep, are you connected? Sundeep?
Yes, sir. He's there.
Yes, sir. I'm connected, sir.
Okay. Do you have presentation? Can you share and go ahead with presentation?
Okay, sir.
If it is there in your system, you can share and present. Dr. Barman, please allow him.
Yes, sir. He is allowed, sir.
But he can skip the introduction part.
Sir,
But he can skip the introduction part. Straight away go to your objectives to save time.
Okay, sir. Namaste. I wholeheartedly welcome all the dignitaries to my thesis presentation. I will begin with the quote, "Engineers design the world, doctors cure the world, but I take pride in saying we the agriculturists feed the world." Today's world is facing many challenges. Among them, a major threat to food security is malnutrition. Pulses play a significant role in mitigating this threat. Rightly, they have been called as the poor man's meat. Black gram, an Asiatic Vigna species, is truly a potential and versatile pulse crop. But black gram is highly susceptible to Callosobruchus maculatus, the larva being the sole damaging stage feeding on the starchy material of the seed. Its occurrence is widespread around the globe. Bruchid damage in pulses like black gram is manifold. The loss in seed weight and protein content has been reported to vary from 45 to 60% in black gram due to bruchid infestation. The lack of detailed bruchid resistance evaluation coupled with narrow genetic base has resulted in scarcity of reliable resistance sources against Callosobruchus maculatus in cultivated black gram. So the need of the hour is to identify novel and natural resistance sources and widen the available genetic base, which will facilitate introgression of resistance genes. There are various approaches to manage or control storage pests like bruchids, but host plant resistance is the relative amount of heritable qualities possessed by the plant which influence the ultimate degree of damage caused by the insects. So how to achieve host plant resistance? The answer lies in plant genetic resources, which are the genetic material of actual or potential value for food and agriculture. With this background, I took up my research work on assessment of genetic diversity and differential reaction of black gram, germplasm and its crop wild relatives against pulse beetle, that is Callosobruchus maculatus. The plant material consisted of 55 land races, 10 crop wild relatives and 4 chicks. We have selected accessions from diverse geographic origin and morphological attributes from the Indian National Gene Bank. This is a glimpse of variability present in the accessions. So the first objective was to assess the genetic diversity of the accessions using microsatellite markers. The DNA was extracted using modified CTAB method given by Doyle and Doyle in 1990. The DNA was quantified followed by PCR amplification and product separation. Let's come to results. 8 highly polymorphic markers have been shown along with their forward and reverse sequences with PIC values more than 0.5. This is the gel image of VR032 locus. The phylogenetic tree grouped the accessions into 3 clusters, cluster 1 being the major, which included the wild and all the check accessions, cluster 2 and 3 contained 2 accessions each. The genetic diversity parameters revealed considerable genetic variability among the studied accessions. The second objective was to evaluate differential response of black gram and its seed developer germplasm to pulse beetle, that is Callosobruchus maculatus. The accessions were screened against bruchid under artificial infestation condition following no choice protocol given by Giga in 1995. Freshly emerged adults were released into perforated bottles at the rate of 2 pairs per replication, and observations were noted for oviposition, exit holes and other growth parameters. The insect parameters like adult emergence, development period, seed weight loss percentage and growth index were calculated using displayed formulae. The qualitative seed traits were assessed as per IBPGR descriptor. In quantitative parameters, seed hardness was measured in texture analyzer using pressure exertion method and measured in Newton. Let's come to results. In ANOVA for growth parameters, growth index showed highest variability followed by adult emergence. IC259504 was observed to be immune and IC424616 was resistant to Callosobruchus maculatus attack based on growth parameters. Considering the hidden infestation of bruchids, X-ray images were captured before and after infestation, thus establishing IC259504 as immune and IC424616 as resistant genotypes. Based on 3 key growth traits, adult emergence, seed weight loss percentage and growth index, accessions were categorized into 6 classes: immune, resistant, moderately susceptible, susceptible and highly susceptible, and IC259504 was immune and IC424616 was resistant on all the 3 parameters. Accessions also possess significant variability in terms of qualitative seed traits. Black was the prominent seed coat color, followed by mottled brown, chocolate green brown and light green. The majority of the accessions had a ovoid shape. About 88% accessions were smooth textured. Similarly, most of the accessions were dull in appearance. In quantitative seed parameters, the 100 seed weight showed highest variability, followed by seed hardness. The scatter plot shows linear relation among 3 key growth parameters. In correlation heat matrix, it is evident that seed weight loss percentage was significantly positively correlated with adult emergence and exit holes and negatively correlated with development period. Growth index had significant positive correlation with adult emergence and seed weight loss percentage. Negative correlation of seed hardness with adult emergence and growth index indicated more hardness of the seed, more force required for emergence. Positive correlation of oviposition with 100 seed weight suggested beetles preferred egg laying in surrounding with more food availability. Further, the exit holes was found to be a better criteria for assessing resistance or susceptibility. So the first key finding was IC259504, a Vigna vexillata accession collected from northern parts of Western Ghats, was immune, and IC424616, a landrace accession of Vigna mungo from Khunti district of Jharkhand, was resistant to Callosobruchus maculatus infestation. Then adult emergence and growth index were negatively correlated with seed hardness. Then exit holes was a better criteria to assess resistance or susceptibility. Then 8 highly polymorphic markers have been shown with PIC value more than 0.5. So what lies ahead? The identified immune and resistant accessions could be utilized for pre-breeding to develop bruchid resistance cultivars in blackgram and related species. The biochemical and molecular mechanisms of bruchid resistance need to be unveiled. The role of other protein and non-protein inhibitors which confer resistance to blackgram against bruchid needs to be studied. Our research article has been accepted in Indian Journal of Plant Genetic Resources with NAS rating of 5.54, another communicated to Plant Genetic Resources with NAS rating of 6.87. A popular article has also been published in Kerala Karsakan from Department of Agricultural Development and Farmers Welfare, Government of Kerala. I have been awarded Best Poster Award at an international conference at AgriVision 2021. I have been nominated for prestigious Dr. K.L. Mehra Memorial Award by Indian Society of Plant Genetic Resources. I have won Shining Star Award at AgriVision. Besides, I have also been awarded Merit Certificate for attending workshops and training programs. I have also presented my poster at 2nd International Agro Biodiversity Congress and delivered oral presentation and participated in various quiz competitions. I have qualified ICR NET and secured All India Rank 2 in ICR SRF examinations. I would like to acknowledge my chairperson and advisory committee, Division of Plant Genetic Resources, PG School IRI, ICR Junior Research Fellowship, my parents and friends. At the end, I would like to quote Melaku Varidi, a renowned geneticist and agronomist. Plant genetic resources are seldom raw materials. They are the expression of the current wisdom of farmers who have played a highly significant role in the building up of world's genetic resource base. Thank you.
May I invite Dr. Bansal for starting the interaction with the student, please?
Okay, Mr. Sundeep Kumar.
Yes, sir. Good afternoon, sir.
Its objectives were clear. Good afternoon. Of course, I will not ask you that what was the basis of selecting these different accessions, and whether there was already through the literature some information available with regard to the susceptibility to the particular storage pest you're talking about it. I think that's the first thing I would like to ask you, whether already some information was available with regard to the susceptibility of these accessions against the particular storage pest you're talking about, or this is entirely a new piece of information that you have generated.
Sir, sir, thank you for asking the query. Sir, the identified immune accession, it was from Vigna vexillata accession. Although there have been reports previously of resistance in Vigna vexillata against Callosobruchus maculatus species, but we have identified a completely resistant accession.
The voice?
Hello, sir.
Is my internet problem or your internet problem, Mr. Deep?
No, it's okay.
It's clear.
It is clear, Dr. Bansal. It is something on your
Anil,
side.
can you hear me, Dr. Anil Dahuja?
Yes, sir. Yes, sir. At overend, the voice is very much clear, sir.
Hello?
Yes, sir. At overend, the voice is very much clear. The Sundeep is completely audible.
Yeah, there's some issue on your side, Dr. Bansal. If you can hear.
Maybe he is not able to hear
Yeah,
us.
yeah, internet has problems. So he was out for 10-15 minutes.
His screen is also freeze now.
Maybe we move forward.
Meanwhile, we can have the comments from Dr. Joshi.
Thank you. Sorry that Dr. Bansal became like a statue, you know. Then we realized there is some problem. Anyway, Sundeep ji.
Yes, sir.
Okay, innovative work, good work. I like it. Previous works were also very excellent. I mean, very nice work. First, I have, normally I ask one question. This time I will ask two questions. First is, how much you are sure that you are saying that material is immune? And second is highly resistant. How much you are sure? 100% sure?
Yes, sir. The immune accession, that is IC259504, it was a bold-seeded accession from Vigna vexillata. We have, this was totally remained uninfested in preliminary as well as in our validation screening, sir. Sir, on that basis, I can say that it is an immune accession to Callicebrucus maculatus, sir.
Very good, thank you. So sorry, I'm asking second question, Dr. Dhillon. Are you making this material freely available to all breeders in India?
Sir, this accession has been identified and path ahead, we can go for registration of this germplasm, unique germplasm. Then we can think of making it available for common use, sir, to develop root resistance cultivars, sir.
Thank you,
sir.
I think now we have Dr. Bansal back. So,
can you hear me, Dr. Anil?
Yeah, yes, sir. Now we are able to hear you, sir.
Okay, sorry, I think there was some problem with internet connection. Okay, I would then come to the second question, Mr. Sundeep, was that how long did you, what was your storage conditions actually, or how long did you store these, seed material along with the particular pest?
Sir, the accessions were screened in a BOD incubator at temperature 65 plus or minus 5 degree and a relative temperature of 28 degrees Celsius and relative humidity of 65 plus or minus 5 degrees Celsius, and it was followed under artificial infestation conditions under no choice protocol, sir.
Okay, that's fine. That's fine. And did you find any correlation between, I think you did some, seed coat structure analysis also. So did you find any relationship between these different accessions, seed coat texture versus susceptibility to this particular pest?
Sir, actually, bruchid resistance, this character is a bit, it is controlled by interplay of various characters of the seed, like it is controlled by the morphology of the seed, biochemical and molecular basis. We cannot, in our study specifically, the oviposition was could not be the oviposition could not be assessed to any particular qualitative character of the seed. So therefore, we on exhaustive literature survey, we found that the number of exit holes are means a reliable criteria to assess the resistance or susceptibility of the accession, sir.
Okay. Thank you. Anyway, I was expecting something different, but nevertheless, thank you.
Thank you, sir.
Any other questions from the other members of the judging committee?
So anyway, I will start. So Dr. Panigri, Mr. Panigri, first of all, thank you very much for working on a very important aspect, and which has implication more for others, so sort of serving the nation through other institutions. But my questions are overlapping with what Dr. Bansal asked and Dr. Joshi also asked. I don't know why Dr. Joshi said, sorry, Dr. Tillo. But anyway, I'm not going to that point. First of all, the title of your work shows resistance to Bruchot. So I would have gone in the presentation first showing how, if the germplasm has been selected on that basis and if it is variable. But you start presentation with this molecular biology or genomics or I don't know, whatever you want to say that, or biotechnology, first from that angle. Then secondly, when you said immune, I was surprised that so much variability within Vigna mungo. I will come to botanical name also later on, but that there I will address Kavita. Probably it seems from publication Kavita is your advisor. That point I will address to her. Then if it is from different species, you must mention that. Later on you told it is some other species. Is it Rajmung?
Sir, the immune accession is belongs to Vigna vexillata, and the resistant accession IC 424616. It is a landrace accession of Vigna mungo, sir.
Okay, that immune accession, does it belong to Rajmu? Yes, Rajmu. What is the vernacular name of that species?
Sir, that is Vigna vexillata tuber cowpea, sir.
So you must mention vernacular name and botanical name. Because when you said pre-breeding, I was surprised. Why pre-breeding? If it is a Vigna mungo, go ahead, do back crossing, use molecular markers. Now we are using a molecular marker very frequently. So it is different species anyway. Then when did you finish your work?
Sir,
Which year?
Sir, first year we went for the preliminary screening. Then the next year we went for the validation screening of promising accession, sir.
Yeah, which year?
Sir, 2020 and 2021, sir.
2021, when I was director, unfortunately, two director and we are sitting there, so asking you hard questions. We simplified registration so much. Only 1 year data are required. And that too, we started accepting whatever is given in the annual report of the institute. So why you are delaying registration and distribution of the germplasm to other pulse breeder? Thereby, you are dividing the nation of the advantage of these good genotypes. My request to NBPGR, Dr. Veena Gupta also seems to be there. Kavita is also there. Celia also I saw the name. Then hardness. Did you see cooking quality?
No, sir. The cooking quality aspect of this accessions were not studied.
I don't know what is Hindi. In Punjabi, we call it kodku. If it is rajmash, rajmung, then there are many kodku. So that's another point which you should take care of. Then my question to my colleagues, not question, just I will say request now, because now I left in the year 2005. When I joined, we have problems. Somebody was talking black gram, other was Urduveen and mash and moong, etc. And I requested Roshni to standardize. We didn't use the word black gram there. Urduveen, somebody was writing in 2 words, somebody in 1 word. And let me share with my colleagues from NBPR now when I see on ResearchGate, most seen of my publication is that I will say just a notebook, etc. Vernacular name, standardized name. So please let us stick to them. Black gram is again Hindi mein kale chane. It is very common. So we decided at that time, and again we are shifting to these. Would have been 1 word, 2 word. So mash, what you want to write or ma Punjabi we call it ma. So that is my request to my colleagues. Please follow that terminology. I'm requesting that. That's fine. Thank you, Dr. Banigri. Please try to register it and then.
Chairman, sir, just one small intervention. You made out very good point regarding delay in registration, but somehow in some of the registration committee meeting, this brilliant idea was given by some committee members that unless we have multi-location data published in form of peer-reviewed research paper, it cannot be registered. And as you rightly said, that this delays the access of material to the people who want to quickly use it. So maybe in your personal capacity, you can write also and see this needs to be revisited.
I will be very happy to do that. Let me share the information, the basis. I did not allow 1 year testing to be listed in the report of NBPGR. I said it should be either 2 locations or 2 years. 1 year testing under natural condition doesn't mean anything. And same thing at PAU, in annual report of PAU. But it started with the mango, Dr. Hargobind Singh, Dr. H.E. Singh, who did. No, is he there or not? He was one of my mentors. And he has brought to the notice one mango variant. He submitted that data and nobody was prepared. And then I spotted that mango tree is there, you go and see it. And we registered that. So we have to be more liberal. This registration is not that. TPFR is entirely different. So I will be very happy to write. Please.
Sir, we have one more hand raised. So can we take up that question?
Sure, why not?
Dr. Yasin. I don't know whether it is by mistake or.
That's how wisdom come through discussion. That's what I feel.
Dr. Yasin, do you have any question?
Sir, actually now for registration, they need 4 years data.
Anyway, what I can say. Why 4 year data?
Okay, then we'll.
It means it means in case of mango, from where it started, you need 10 years to register something, because flowering will come after 5 years.
Sir, I don't know, sir. I have submitted some material for registration, but they said without 4 years data, it cannot be accepted. So my application came back.
You are from NBP, from which institute, please?
Yes, sir. From NBPGR, sir.
Dr. Veena Gupta was very much party to that decision. Dr. Kumar was party to that decision. I think you discussed with them. And
Chairman, sir, I think we can take this discussion part
later on. Yeah.
So with this, now we'll move to the next presentation, which is by Ms. Shravya Singh from Discipline of Plant Pathology.
Shravya Singh.
Good afternoon, everyone, and welcome to my presentation. 2019, the year when coronavirus pandemic brought huge challenge for us, shattering lives across the world and still haunting us in how. It showed that how a piece of RNA particle is able to completely change and reshape the world and cause a havoc across. In this mountain of despair, a stone of hope was found in the form of vaccination, in which a mild, killed or inactivated strain could fight with the severe COVID and result in dead or inactivated COVID. So the vaccination with mild or inactive virus was used for immunization protection for fighting against COVID. Plants also suffer from many diseases caused by bacteria, fungi, viruses, and nematodes. Out of this, viral diseases pose a major challenge for plant health protection. So if we are immunizing people for fighting against COVID, so why not immunize the plants the same way as vaccinate the people? Why not develop pre-immunized agricultural crop plants against the viral pathogens? The answer for this is development of superinfection exclusion or cross-protection system for management strategy for controlling viral diseases. Citrus, the third most produced fruit crop of India, is affected by many diseases, out of which Citrus stagia is the major disease in the country. It is caused by a virus called the Citrus stagia virus, which produces symptoms such as vein flecking, vein corking, seedling yellow, stem pitting, and citrus decline or death of tree. It is a phloem-limited aphid-transmitted infectious virus, and the genome of CTV is having 13 ORFs, of which the P33 protein in 3' half is responsible for superinfection exclusion. In India, the CTV incidence is present in all the citrus species and in all the citrus growing regions of the country, but the maximum incidence occurs in the northeast region with maximum 75% incidence. So what is basically the threat? The threat is the failure of traditional management practices, the abandonment of old citrus orchards which are affected by citrus decline, and the spread to new healthy orchards. And the approach for management is the development of mild strain cross-protected immunized citrus planting materials. There's a huge biological and genetic diversity of CTV genotypes in India, and the maximum diversity is found in the northeast India. So with this much diversity, the occurrence of mild strain and natural cross protection is obvious. Thus, we need to search mild strain that will be exploited for the CTV decline management. History of cross protection in CTV research has been that it was initiated in the country by Kapoor and Rao 1967, Kapoor and Chakravarti 1980, and Balaraman and Ramakrishna 1977. But it failed. It failed because it was only based on the symptomatology and aphid specificity. There was no inclusion of phylogeny. There was no inclusion of the relatedness. In Australia, Brazil, South Africa, and Peru also mild cross-protecting systems have been developed for CTV, but they have not came into the commercial use. So why to go for cross protection as a management strategy? The answer is that there is a lack of availability of disease-free planting materials. Eradication is not possible since it is very time-taking also, and the transmission is way much faster than eradication. Third reason is that there is a low success to obtain transgenic resistant plants. So MSCP, which is the mild strain cross protection, it is a process in which we introduce a mild strain in a plant to protect it from the severe strain of the same virus. Folimonova et al. 2020 indicated that it is the result of the genetic traits of mild and challenging strain, and there should be a homology between the mild and severe strain to ensure that it is a mild strain. So me, Shavya Singh, I have completed MSc degree program from the Division of Plant Pathology in year 2021. I am presenting for the IRI Merit Medal Contest, and my research title was Identification of Putative Mild Cross-Protecting Strains of Citrus Stagia virus for Management of Severe Decline Disease in Citrus. The objectives under my research program were: first, determination of genetic variability of Citrus Stagia virus and identification of the predominant strain in Northeast India in current condition. The second objective was identification of putative mild cross-protecting strains of CTV through in silico codon uses biasness analysis. So under the first objective, where the aim was identification of dominant CTV genotypes, the following activities were performed. The collection of isolates were done from citrus orchards of Northeast region, and the disease incidence was estimated from it. So the overall CTV incidence was found to be 65.7% with a maximum of 71.4% in HRS farm Assam. Even 100% disease incidence was found in many cultivars, and the maximum infection was found to be in the rough lemon among all the cultivars. 12 out of 21 isolates were then taken for PCR amplification and gel observation, and they were maintained in citrus host by wedge grafting in the greenhouse of Advanced Center of Plant Virology in the Division of Plant Pathology in the campus. Molecular cloning of ORF1A gene fragment was done, and after the positive clone confirmation, all were sent for sequencing. The phylogenetic tree was constructed using MEGA6 program for the studied isolates along with other Indian and international isolates also, and it was found that the isolates segregated into total 10 genogroups. But the present isolates segregated only into 2 genogroups, out of which the majority came into the VT-KPG3 group. Intra-farm genetic diversity was also observed in the present study. So the recombination analysis was also performed using the program RDP4, and where 6 isolates were found to be the recombinant isolates and they were related to VT-KPG3. So the final conclusion of objective 1 was that there was dominance of severe CTV strain VT-KPG3 in the Northeast India. So the following activities were performed under the objective 2, where aim was the identification of the mild type strains. So these are the source of complete CP gene sequences used for curve analysis, in which 12 isolates were taken and 5 reference isolates were also taken. Was plotted against the GC3 values of CTV-CP gene. It was observed that all the points lie below the standard curve that indicates that codon choice is influenced more by selection than the mutation. The composition analysis showed that CTV-CP gene is biased for uracil-ending codon, which is the same as the citrus host. So it is confirmed that CTV as well as the citrus host showed the codon uses biasness. In the GC12 versus GC3 plot, also it was observed that it followed a unique pattern of higher GC1 and GC2, same as its host. So it indicates finally that the codon biasness of CTV is more dependent on the host tRNA pool. In the following graph, we can see that the ENC values are there for different isolates. For an isolate to be mild, its ENC value should be lower. So only 3 Indian isolates fulfill this criteria, which are K10, MNP1 and MB3, which were having lower NC values. At last, all isolates were grouped based on codon biasness, phylogeny of CP gene, phylogeny of ORF1A gene, and recombination event, and it was observed from the comparative analysis that K10 was distinct based on both the 5' ORF1A and CP gene. MNP1 was genetically not so closely related with VTKPG3. However, it is a non-recombinant isolate, and MB3, it was genetically very close with VTKPG3 isolates, and it was also a non-recombinant isolates. So the conclusion of objective 2 was that isolates K10, MNP1 and MB3 are mild strains, but MB3 may be a stronger mild strain. Finally, the biological indexing was done for in silico based mild CTV isolates, and all the 3 isolates produced mild type symptoms on different citrus hosts. However, the challenging with severe strain could not be done due to the limited time frame of MSc program, but I would like to further continue this work in my PhD degree program in the division. So the final conclusion of my research work are that overall CTV incidence was found to be 65.7% in Northeast India, and VTKPG3 was found to be the most dominant severe CTV strain there. K10, MNP1 and MB3, these 3 were found to be mild type isolates from codon biasness analysis, and the phylogeny also confirmed its genetic similarity with the severe CTV strain VTKPG3. Through biological indexing, it was confirmed that all these 3 isolates were showing the mild type symptoms. So the utility of my research work is that it could be used for development of pure and effective mild strains, and it can be used for production of immunized planting materials which could be supplied to the growers of Northeast India and result in the upliftment of socio-economic status of that region. 2 research papers were submitted, out of which 1 is accepted, and I have received 1 Best Poster Award, given oral presentation for Professor M. J. Narasimhan Award, served in PGSSU 2021 as class representative for MSc, and having life membership of Indian Phytopathological Society. I have got ICR, JRF and SRF fellowships, and have participated in several literary and cultural activities during my MSc program. I would like to thank Chairman, other members of my advisory committee, lab members, Professor of the division, Head of the Division of Plant Pathology, all the faculties, Dean and Joint Director PG School, and ICR for PG Fellowship. I would like to thank all of you for giving me the platform and opportunity to present my research work. Thank you.
I would invite Dr. Joshi to please start the discussion.
Yes, thank you. I raised hand because I like the way the research has been planned. There is a story. See, I'm not a citrus or don't work in fruit vegetables, but I like the story. That's why I raised a hand. That we all should do research in like a storyteller, after some time we can tell step 1, step 2, step 3, step 4. I was also glad that it was told that, of course, time was short. So she said that I will be happy to do that. So congratulations, Savia, for your whole team. My question is, you said that time is short. So I did in silico study. How much you are sure if somebody or you continue to do the same thing in the field condition, how much the result will match?
So the in silico analysis was done because in 2020 only, in January, a paper was published by Folimonova in which he indicated that there should be a genetic similarity also with the severe strain to be qualified as mild strain. And the purpose of codon usage biasness analysis came from there only. So it was a new point to explore, and thus I have taken that.
And after that in silico analysis, the 3 isolates were found to be putative mild type strains, and they were also biologically indexed, and they were showing mild type symptoms, as well as from where they were collected, there also they were showing mild type symptoms only. But the challenging experiment, it requires more time, and because in 2 years program, many months have gone in lockdown, and we were not in campus, and citrus requires more time for challenging and then again proof concept experiment. Thereby, I have done only the biological host indexing and in silico analysis.
Thank you. In lockdown, we are speaking more. Thank you.
May I now invite Dr. Bansal for the interaction?
Okay, Ms. Shavaiya, I think an excellent piece of work.
Thank you, sir.
And an excellent presentation too.
Thank you.
Dr. Joshi mentioned, I think it was very clear, and that's what expected of students from IRI. In fact, in other disciplines as well. Of course, they are doing all the best. In fact, given the opportunity.
Is internet working?
And anyway, coming down to the work you presented, of course, very clear. But point is that you found 3 mild strains you mentioned. You think there'll be more? And what is the probability of occurrence of these mild strains versus severe strains? What is your prediction from the region you're talking about? It for citrus grown is very, very important. I think the world's old century of, what is that, kind of collection is also there in situ, grown in North Hills of citrus, if you know about it.
Yes, sir. Sir, for that only I have taken objective 1 to find out that what is the major severe CTV strain that is there in Northeast India conditions. And all the strains that were found to be severe and decline inducing were found to be related with VT-KPG3. So VT-KPG3, that is in Israel isolate VT, and which is related to Kalimpong isolate KPG3. They both are related. So on the taking it as a reference, whole research was planned. So all the 3 mild strains, they are they are developed only against that VT-KPG3 and the related isolates only. For this purpose, only first objective was taken to determine that what is the most dominant isolate which is available in Northeast India in the current conditions.
And thank you. And I hope you'll be able to continue this good piece of work. I mean, I don't know whether you're there as a PhD student or whenever you get an opportunity, must pursue it hard.
Yes.
All the best.
Thank you, sir.
Just a small query from my side. Excellent study again, hearty congratulations. And
thank you so much.
Very clear message you gave, and you spoke very well, my query that you have worked for the northeastern region. What about the other regions, Central India or wherever these citrus are quite successfully being grown? Whether the same phenomena, same hypothesis will work the other places?
So there were 2 reasons for selecting Northeast India. When I thought that I will be working on CTV, then there were 2 reasons. First of all, that the genetic diversity of citrus is predominant there only in Northeast India, as well as the disease incidence of CTV is also maximum there. And more the genetic diversity, so the occurrence of natural cross protection and the naturally mild strains, it will be the probability of occurrence of mild strain will be more in the northeastern conditions. That's why I've taken that. But so I have done in northeastern conditions only in my MSc program.
Okay. That was nice. Thank you. All the best.
Thank you, sir.
Oh, may I, Dr. Tuja?
Yeah, sir, please.
Yeah, just to save time. Ms. Seva Singh, very good presentation. Everything very good. But you're lucky to work in a wonderful lab on virology. Everything is very good. But how about good publications? Where is the link? My question is related to what Dr. Joshi asked. How much, I am not a virologist, how much virologists are confident or have faith in your work? But at the same time, when we say failure, that is also a step towards success.
Yes.
Research, it is no research if there is no failure. Then it is repetition. So was something missing somewhere? Why didn't you publish in high quality
Sir, I have submitted in 3 biotech, but it is taking time. I have revised once. I think 1-2 more week it will take. And in Indian phytopathology, I have published.
Indian phytopathology तो home journal है.
Yes, sir. Sir, in second journal also I have communicated, but
Anyway, I mean that would have given more confidence to us that you are on path to success. You are on correct path. Whether results are good or not, you are on correct path. But success and correct sometimes they differ. Anyway, thank you.
thank you, sir.
Thank you, sir. So next presentation in the sequence is from Mr. Sinto from Discipline of Plant Physiology.
Namaste everyone. I am Sinto from the Division of Plant Physiology. Today let's talk about climate change. People are calling it the crisis of our time. Various anthropogenic activities like burning of fossil fuel, deforestation, certain agricultural practices like excessive use of nitrogen fertilizer have significantly contributed to the greenhouse gases. And the result is a rise in Earth's temperature. Since the dawn of Industrial Revolution, CO2 levels have dramatically surged from 280 ppm to present-day 418 ppm, and it is expected to reach 650 to 1200 ppm by the end of this century. Thus, elevated CO2 levels will influence crop performance. Wheat contributes to 27% of global grain production, but wheat cultivation will face significant challenges brought about by climate change. When wheat is grown under normal condition, there is continuous uptake and loss of water in the form of transpiration. Whereas under elevated CO2 condition, there is closure of stomata and reduction in transpiration, which in turn increases the canopy temperature. The mass flow is also affected, affecting nutrient allocation, thus reducing the concentration of 25 mineral nutrients by 8%. The nitrate uptake is reduced under elevated CO2 condition, followed by limited nitrogen remobilization to grains, thus reducing the concentration of grain nitrogen by 6 to 16% and grain protein content by 10 to 15%. Elevated CO2 also induces altered phenology, which changes flowering and senescence pattern. Can changes in crop performance and grain quality be attributed to CO2 memory? A recent paper by Dyer et al. In 2019 showed that elevated CO2 enhanced the post-anthesis nitrogen uptake in wheat. Can this be utilized to rescue the grain protein decline? To answer this question, we postulated a study with the title Interactive Effects of Elevated CO2 and Nitrogen Fertilization on Yield and Nitrogen Response of Bred Wheat. Two objectives were put forth. First, to study the effect of reproductive stage nitrogen application on grain yield and nitrogen use efficiency of wheat genotypes under elevated CO2. And second, to study the effect of elevated CO2 and nitrogen interaction on the source and sink gene expression. The first experiment was conducted in Rabi 2020 in the open-top chambers of the Division of Plant Physiology IRI. 6 genotypes were used in the study, 4 high-yielding genotypes and 2 genotypes contrasting for nitrogen efficiency. 3 doses of nitrogen was provided, low nitrogen of 30 kg per hectare, optimal nitrogen of 120 kg per hectare and high nitrogen of 150 kg per hectare. The nitrogen was applied in 3 split doses, as a basal dose, at pooting stage and at 50% anthesis stage. High nitrogen treatment had an incremental application of nitrogen at the booting stage and at 50% anthesis stage. Two levels of CO2 were maintained in OTCs, ambient CO2 of 400 ppm and elevated CO2 of 700 ppm. We observed that under elevated CO2 condition, grain nitrate content significantly reduced by 9 to 50%, while leaf nitrate increased by 81% and stem nitrate increased by 52%. We also observed that the grain phosphorus content increased by 26 to 67%, whereas grain potassium content decreased by 13 to 37%. Grain calcium content varied inconsistently. The grain iron content had also significantly increased by 68%, with higher expression of YSL6 gene, which is known to be involved in iron remobilization to grains. Thus, we found that grain nitrate showed significant reduction, while leaf and stem nitrate increased under elevated CO2. Grain potassium content reduced, while grain iron content increased under elevated CO2. Genotypic responses to elevated CO2 was variable. BTS, a high nitrogen use efficiency genotype, was significantly impacted and was selected for further evaluation. BTS was grown in the open-top chambers of the Division of Plant Physiology IRI. An additional nitrogen treatment was included with 135 kg per hectare. The incremental nitrogen of 15 kg per hectare was provided only at 50% anthesis stage, and the seeds from previous season were used for sowing. The observations were recorded at the following developmental stages: at mid-vegetative stage, at 50% anthesis stage, 15 days after anthesis, and at physiological maturity. It was observed that ACON showed early seedling emergence while ECON showed delayed seedling emergence. The plants under elevated CO2 showed 2 to 7 days delay in heading and anthesis stage. There was a significant reduction in variable and maximal fluorescence under elevated CO2, whereas no change in Fe by Fm ratio was observed, which is indicator of the efficiency of photosystem 2. Size of the flag leaves was reduced under elevated CO2 conditions. Leaf area and dry weight were higher in high nitrogen and high reproductive stage nitrogen. Under elevated CO2, there was a 1.42 to 1.95 degree increase in the mean canopy temperature. This was supported by an increase in leaf area and decrease in stomatal conductance under elevated CO2 conditions. High nitrogen enhanced the production of ROS. Surprisingly, there was a lower accumulation of hydrogen peroxide under elevated CO2. This was supported by an increase in catalase activity under elevated CO2, especially under high nitrogen supply. We also observed that there was a decrease in primary assimilation by GS and GOGAT in flag leaves. Surprisingly, there was an upregulation of GS and GOGAT isoforms which are involved in nitrogen remobilization in high reproductive stage nitrogen treatments. Although no significant difference was observed in nitrogen utilization efficiency and the amount of nitrogen remobilized to grain, we observed that post-anthesis nitrogen uptake had significantly increased under elevated CO2 conditions. We observed that there was no reduction in grain protein content under elevated CO2. High reproductive stage nitrogen application under elevated CO2 increased grain protein content by 1.96%. There was also upregulation of NAMB1 transcription factors which are associated with grain protein content. Elevated CO2 generally enhanced 1000 grain weight and yield, but a 19% reduction in 1000 grain weight and 15% reduction in yield was observed in EC grown plants receiving high reproductive stage nitrogen. This was supported by the reduction in grain size under elevated CO2 condition. Under elevated CO2, the grain length increased by 3.39 to 8.17%, whereas the grain width reduced by 0.93 to 16.54%. There was a higher expression of genes related to grain length and grain width under high reproductive stage nitrogen, whereas the expression of these genes reduced under low nitrogen supply. Thus, we observed that exposure to elevated CO2 in previous generation affected seedling emergence and phenology in Rabi 2021. The accumulation in flag leaf was lower in elevated CO2 despite high nitrogen or high reproductive stage nitrogen supply. Elevated CO2 increased the grain length by upregulating TAGL3 and TAGL7. Elevated CO2 grown plants had higher post-anthesis nitrogen uptake and no reduction in grain protein content when additional nitrogen was provided. I would like to conclude by saying that the reproductive stage nitrogen application is a potential strategy to elevate the decline in grain protein content under elevated CO2 condition. Observations on different physiological parameters indicates that very high nitrogen supply was imposing stress, while high reproductive stage nitrogen did not. The multi-generational exposure to elevated CO2 will have major implications in crop establishment and performance as observed in Rabi 2021 experiment. Thus, considering the variation in genotypic responses and the utility of high reproductive stage nitrogen treatment, the fertilizer regimes need to be revised carefully for future elevated CO2 conditions. The possibility of strategizing current nitrogen dose into more effective split doses and foliar fertilization is promising. The altered crop phenology and associated variation in nitrogen consumption is another important arena to explore. Currently, 2 of my manuscripts have been accepted with a NAS score of 6.55 and 5.23. My third manuscript has come for revision in a NAS score of 9.72. I have also contributed to 2 book chapters. I have been awarded the Young Researcher Award by the Indian Society for Plant Physiology. I have also secured first and second place at quiz competitions organized by National Conference of Plant Physiology. I have submitted 4 sequences related to grain length and grain width to NCBI and have delivered 2 oral presentations. I have participated in poster presentation at NCPP 2021 and in a science quiz organized by Xrd Tirupati. I would like to thank my chairperson Dr. Lakshmi Sati, my co-chairperson Dr. Vishwanathan Chinusamy, Dr. Madanpal Singh, Dr. Shailendra Jha for the guidance and facilities without which the research could not be completed. I also like to thank all the members of Lab 204 and the members of Division of Plant Physiology, my family and friends. Sincere thanks to ICR for providing me with
किधर जाना उसने
information
कुछ बताया
and
था आपको?
IRI for molding me into what I am today. I would like to end my presentation by quoting the words of German ambassador Walter Linde. Every fifth person on this planet is Indian. Without Indians, you cannot solve any big world problems, and one of the biggest is climate change. Dhanyavad.
I first invite Dr. Bansal for the interaction with the student.
Yeah, thank you. Thank you. Your name is Mr. Sinto, right?
Yes, yes, sir.
Okay, I think a good concept to combine the positive aspects of both elevated CO2 and nitrogen. But my question is, of course, related to, you did not do any measurement or estimation on stomatal conductance, though you talked about it initially. I think which was important to relate your results, finally also with stomatal conductance is a very important parameter. You went down to the level of measuring some of those, gene expressions related to certain parameters, you talked about it. But I think pure physiological analysis alone, could have given you deeper insights into the interaction of these two, important, compounds, carbon dioxide, or even, for example, nitrogen. That's one. Second is that, you also did not, I believe, if I missed, please tell me, any measurement on root aspects of it. Because ultimately, when applying nitrogen, you mentioned at the reproductive stage, that time roots may not be as active to take up the application of, any kind of fertilizer that matter, not only nitrogen. So how you think that the roots would have become active, or did you take any measurements to support your results further? So these are my two clarifications actually.
Sir, for first question, sir, like first clarification, I would like to say that I have showed the data on stomatal conductance in the second season. Like it was recorded using Lycor 6400 XT, and we have also recorded other parameters of photosynthesis and transpiration, but it was not shown. But stomatal conductance was shown in regard to canopy temperatures, and like we didn't do any additional like on root parameters, sir, because since it was grown on pot culture. So like we had to either sacrifice few plants for root study. So we couldn't conduct on root parameters.
Since it was pot culture only, I asked you a question. I won't have
Yes.
I understand that, but also the point is, anyway, we'll see that later, discussion part of it. But also you measured, I saw in your presentation, one of the slides, 418.09 ppm. As on 27th of January 2022 of CO2. Did you measure it?
No, so it's actually from NOAA, like from Mauna Loa, what they have measured, sir. That is the present Earth ppm of CO2.
So you don't know how to measure yourself?
No, we have measured in the chamber, sir. Like yes, sir. We used two methods. So one was IRGA was like 6400 XT. We can use the IRGA head for measuring. Another, we used this CO2 sensor from Vaisala, sir. It was also used to measure the CO2 level, constant CO2 levels of measured.
Okay, I will come back if there are more clarifications required. So, thank you.
Thank you, sir. So may I now invite Dr. Patra for the interaction?
Yeah, thank you. Mr. Shinto, a new subject. In the context of climate change, when the elevated carbon dioxide is there, so we say carbon fertilization. So obviously, there is expectation that growth will be more. And also, if the other resources are not limiting, it will improve, it will increase the biomass productions. So in this context, the importance of nitrogen, obviously, otherwise also in normal soils also, nitrogen is always a key nutrient for the production. So I understand you must have applied soil, or it is foliar application of nitrogens.
The soil application.
Soil application of nitrogens. And in this, the uptake was more when we applied at the critical stages. And you are mentioning the nitrate nitrogen in the leaves, in the stem. Is it nitrate nitrogen or nitrogen?
So it was grain nitrate, leaf nitrate, and stem nitrate in season 1 was calculated.
Nitrate or nitrogen?
Nitrate. Sir.
But normally we calculate nitrogen.
Yes, sir. We also calculate nitrogen, sir. But I have showed the data of nitrate, sir.
What was the purpose?
Sir,
because we normally do not do like that.
Sir, actually, there are recent reports where that it says that germination, seed vigor can be affected by grain nitrates. Sir. So we also observed in second season that there was changes in emergence rates, as I showed, under elevated CO2. So, like under elevated CO2, there is decreased grain nitrate and delayed emergence, seedling emergence in season 2. So that was a bridging gap between season 1 and season 2.
And also, if I may come here in between with the chairman's permission, how you expect the nitrate to be present in leaf and stem and in grains? Is it not the reduced form as he's asking of nitrogen to be present there? Since the machinery is there to reduce the nitrate.
Yes, sir.
Yeah, it will not remain as such, for
the moment it enters the root, right?
For a short time, when you too much nitrogen you apply, sometimes it remain nitrate, but otherwise, all will be reduced. The protein form, you are after all estimating the, the proteinous form of nitrogen.
Yes, sir.
And the last thing, a lot of things we know, and also the participants, they are knowing about this. What was exactly new in your study, which was not known earlier?
So we found that there was under elevated CO2, those expression of genes were previously not reported. So we found that there was expression of genes like NAM B1 transcription factor they had enhanced, and also even YSL6 had enhanced the expression under elevated CO2, and even grain length and grain width when the nitrogen is applied at post ANSYS stage, so it is not much studied. So that we had and related gene expression also was studied. So.
Okay, thank you. All the best.
Thank you, sir.
May we have the comments from Dr. Joshi?
Yes, thank you, Shinto-ji. Good presentation. I think you were enjoying your own voice. So I was happy to see your expression that time. Now, this hypothesis is, okay, hypothesis is good. But can you tell me how much you believe that only CO2 will go up and other things will remain constant?
Sir, along with CO2, as CO2 rises, there will be increase in temperature also, sir. But until we won't study the individual parameters, we cannot judge like how the plant will be behaving in future scenario.
Okay, thank you. Thank you.
May we have the comments from Dr. Patil now?
Yeah. Mr. Sinto,
yes, sir.
Generally there is a elevation in CO2. The leaves, actually, there is a phenomenon, leaves gets thicken. And have you measured the how much percentage increase in the leaves thickness over the normal?
Yes, sir. Like we had measured, only in flag leaf we had measured, sir. Like we had measured in terms of flag leaf area and flag leaf weights, specific leaf area, specific leaf weights.
The thickness of the leaves, how much percentage it will increase above our control one?
So we haven't measured any parameters related to leaf thickness.
But then why leaves thickness increases in the elevated CO2? What is the phenomenon? Do you have any idea?
So we observed that there is increase in specific flag leaf like specific leaf weights, sir. But exact phenomena I'm not aware.
Okay, thank you.
So one minor point again, I'm sorry, coming back. I think, point is that you took 6 different genotypes of wheat.
Yes, sir.
So what was the idea and what information did you drive extra, by taking these 6 genotypes 4+2? Could you relate your results with the different genotypes you could take?
So, like first, initially the high yielding genotypes were taken based on previous data, which was observed from our own division of plant physiology, sir. So, phenomic data suggests that those 4 genotypes has higher nitrogen use efficiency, and our lab has been working on BT Schomburg from quite a long time, sir. And we have based on seedling stage and various field trials, we have shown that it has higher nitrogen use efficiency. So, in contrast, we have taken gluous early as low nitrogen use efficiency.
All right. Over to you, James.
May we have the comments now from the chairperson?
Okay, Dr. Doja. Thank you. Mr. and true. Many of the things or most of the things were expected, but how about emergence? How that is affected by elevated carbon dioxide?
Sir, as I said previously, sir, the rate of germination is affected by grain nitrate content, sir. So we observed that in season 1, the same seeds were sown in the second season. So we observed in the first season, Rabi 2020, there is reduction in grain nitrate content and elevated CO2. And subsequently, next season, when there was emergence, we found that these seeds which had lower grain nitrate had those emerged late, whereas those which were grown to ambient conditions had higher nitrate content, had higher seedling emergence.
I am talking about carbon dioxide.
Sir, even we have predicted that there could be any kind of epigenome, their transcript changes in epigenetic regulation, but we haven't touched those areas. So it will be my objectives under my PhD study, sir.
Okay, thank you.
Thank you, sir. So we can now move to the next presentation, which is by Ms. Sushma from the discipline of Seed Science and Technology.
A very good morning to all the dignitaries who are attending this presentation, eminent scientists, respected teachers, seniors, juniors, and my dear friends. I welcome you all. As we all know about the Thomas Malthus, who gave the theory on population growth in the 17th century, according to this theory, a population is increasing in geometric way while the food production is increasing in arithmetic way. In future, the people will not die because of war, but it's because of hunger. So these food problems have hunted mankind since time immemorial. Then the question is how to overcome this problem. Through expanding the cultivated area? No, it's a limiting factor. The only possible way is by improving the production per unit area. As we know about the quote which is mentioned in the Manusmriti, that is Subijam Sukshetra Jayate Sampadayate, which means good seed in good soils yields abundantly. Then what is quality seed? The seed with high genetic purity and physical purity, maximum germination percentage, minimum enough moisture content, good seed health and maximum seed vigor. So use of these quality seed is one of the way to improve the production per unit area. Cereals and pulses are the major staple food grains to feed the increasing population, and among the pulses, chickpea is the major crop occupying the larger area under cultivation. It has many uses, that is, it has considerable amount of proteins, minerals, unsaturated fatty acids, dietary fibers, beta-carotene, and some amount of carbohydrates. So it can be utilized to make a nutritionally value-added foods, which can then be used to feed the low-income people in the developing countries. In this crop, low yield and seed deteriorations are of the major research concern. So in order to improve the seed quality, there are several seed quality enhancement techniques that have their own benefit. But the applications of nanoparticles is one of the new approach as compared to their applications in the industrial and biomedical sectors. This nanomaterial can be defined as the material with at least one dimension less than 100 nanometer, and due to their small size and greater surface activity, they possess some unique physical and chemical characteristics. Thus, their reactivity enables them to have novel applications in the different sectors such as food, pharmaceutics, textiles, energy, etc. Presently, this nanotechnological application has provoked the interest in the agriculture sector as well. There are n number of studies has been done on the effect of nanoparticles with respect to seed quality and other aspects. But the gap which we found was there was limited information available on the effect of nanoparticles on seed yield and seed quality parameters of chickpea, and there was less information was available on the effect of nanoparticles in improving the seed storability of chickpea. With this, we propose the research work with the title of Effect of Nanoparticles on Seed Quality, Yield Attributes and Storability in Chickpea. I am Sushma from the Division of Seed Science and Technology, contesting for the IRI Merit Medal. Then the objectives of my study was to evaluate the effect of nanoparticle seed treatments on seed quality parameters in chickpea, to study the effect of nanoparticle seed treatments on seed yield attributes in chickpea, and to assess the efficacy of nanoparticle seed treatments in improving the storage potential in chickpea. Material which we used for the study was chickpea variety Pusa 547, and the seeds were sent to TNEU for the seed treatment with a different combination, which consists of different nanoparticles, forms, formulations, and dosage, along with the two control, that is, one is untreated, another one is treated with a recommended POP, that is thiram at the rate of 2 gram per kg. Here is the treatment details, which consists of 62 treatments including two controls. In the first objective, treated seeds were evaluated for various seed quality parameters, and all the studied parameters showed the significant difference among the seed treatments. Here it is the graph showing percent increase or decrease in radical emergence percentage in different treatments when compared to both the controls. Among the different treatments, T15 showed significantly highest radical emergence percentage, that is of more than 10% increase, and T28 showed lowest radical emergence percentage, that is of more than 25% decrease compared to both the controls. And T15 showed more than 5% increase, and T28 showed more than 40% decrease in germination percentage compared to controls. And significantly highest total seedling length and seedling vigor index 1 was observed in T15 and lowest was in T28 compared to controls. And more than 10% increase in seedling vigor index 2 and more than 50% increase in dehydrogenase enzyme activity was observed in the seeds treated with the diurnal zinc oxide 250 ppm compared to controls. And T15 showed more than 30% decrease in electrical conductivity, and T15, T16, T54 showed more than 50% decrease in pathogen infection percentage compared to controls. The conclusion drawn from the first objective was significantly highest radical emergence percentage, germination percentage, total seedling length, seedling vigor index 1 and 2, dehydrogenase enzyme activity, and lowest electrical conductivity were recorded in the seeds treated with the diurnal zinc oxide 250 ppm compared to both the controls. And significantly lowest pathogen infection percentage were recorded in the seeds treated with the diurnal zinc oxide 250 ppm and 500 ppm and diurnal titanium oxide at 100 ppm compared to both the controls. In the second objective, treated seeds were sown in the field with a randomized block design, and observations was taken on minimum of 5 randomly selected plants per treatment per application. Then the observations recorded were field emergence percentage, plant height, number of branches per plant, days to 50% flowering, number of pods per plant, harvest index, seed yield per plant, and test weight. And all the studied parameters showed the significant difference among the seed treatments. T15 showed more than 10% increase, T32 showed more than 30% decrease in field emergence compared to both the controls. And among the different treatments, T15 showed the early flowering compared to controls. And more than 20% increase in plant height and more than 30% increase in number of branches per plant were observed in the seeds treated with the diurnal zinc oxide 250 ppm compared to both the controls. And more than 30% increase in number of pods and more than 80% increase in seed yield per plant were observed in the seeds treated with the diurnal zinc oxide 250 ppm compared to both the controls. And more than 10% increase in harvest index and test weight were recorded in the T15 compared to both the controls. Then the conclusions of the second objectives were all the studied parameters showed significantly highest values except days to 50% flowering, which showed the lowest values in the seeds treated with the diurnal zinc oxide 250 ppm compared to both the controls. In the third objective, treated seeds were stored under laboratory condition, one set of seeds in cloth bag, another set of seeds in polythene bag for a period of 6 months. And stored seeds were evaluated at various stages, that is 0 month, 3rd month, and 6-month period of storage. Results of the germination percentage showed significantly highest germination value in the seeds which were stored in the polythene bag than in the cloth bag, and there was a decreased germination percentage over a period of time. And all the studied parameters showed better quality of seeds in the polythene bag than in the cloth bag. And among the treatments, T15 showed significantly highest germination percentage compared to both the controls. And this T15 also showed significantly lowest abnormal seedlings and dead seeds percentage, significantly highest seedling vigor index 1 and 2, and significantly lowest moisture content and highest field emergence percentage compared to both the controls. And the conclusions of third objective were, after 6-month period of storage, significantly highest germination percentage, seedling vigor index 1 and 2, field emergence percentage, and lowest dead seeds percentage and moisture content were recorded in the seeds treated with the diurnal zinc oxide 250 ppm, which was stored in the polythene bag over both the controls. and significantly lowest abnormal silice percentage were recorded in the seeds treated with the drinano zinc oxide 250 ppm, drinano titanium oxide at 100 ppm, drinano silicon dioxide at 500 ppm, which was stored in the polythene bag over both the controls. Then the salient finding of the study was, among the nanoparticles it was zinc oxide, and among the forms it was nano, and among the formulations it was dry, and among the dosage it was 250 ppm, found to be the most effective treatment for the enhancement of seed quality, yield attributes and storage potential in chickpea. The enhanced performance in the zinc oxide nanoparticles treated seeds could be due to the rapid water uptake in the treated seeds through the nanopore formed by the nanoparticles, and this nanoparticle stimulates the GA biosynthesis, thereby the increased enzyme activity and starch hydrolysis. This leads to the rapid embryo growth, and it also enhances the germination through the cell wall loosening and endospore weakening. These studies showed that use of nanoparticles will improve the germination and uniform stand establishment at field level, and by using these nanoparticles, numerous metabolic, biochemical and physiological processes can be unlocked to increase the potential of the crop for the better returns to the growers. Future line of work is detailed biochemical molecular studies need to be done on underlying mechanisms of nanoparticles' effects on seed quality. My few achievements are, one of my paper has been published in the International Journal of Tropical Agriculture, and one paper has been accepted for the publication in the Seed Research Journal, and one paper is in the advanced stage of review in the Turkish Journal of Agricultural and Forestry. And one review article has been published in the Seed Research Journal, and one manual chapter has been published, and I have received Junior Research Fellowship and qualified ICR NET and ICR SRF with All India Rank 6. I've also participated in the national conferences, webinar and cultural activities. And I am happy to inform you that I have received 15 gold medals from the University of Horticulture Science Bagalkot during my undergraduation program. Finally, I would like to thank everyone who has helped in making my research a successful one. In winter steals our summer seed, a farmer's work so that world can eat. Therefore, it's high time that we all join our hands together and work for the betterment of farmers, the lifeline of our nation. With this, I, Sushma, sign off. Thank you.
May I invite Dr. Joshi to start the
Thank you. Thank you very much, Sushma ji. Very nice presentation. Theme is also very good. Theme is also matching with the future requirement, nano, you know. But your results are also very good. I should not ask this question to you. You are only masters, you know. Some voice is coming, free voice is coming from somewhere. Please mute, I think. In lockdown,
you are muted.
You're muted.
Sorry, I was muted. Sorry. So somebody muted me also. I was unmute. But this is the power of technology, Dr. Dhillon. You are playing some magic. Thank you. So Sushma, everything very nice. Now, this question, I'm hesitating to ask you, but I'm asking you because zinc, you have found zinc oxide as something very interesting in nano. And whenever we do experiment with zinc, we have to be very careful. So the question is, did you map your field by doing soil testing for variation on these molecules which you are going to use? You please unmute yourself.
Sushma, please unmute yourself.
Yes, sir. No, sir, we did not study any soil. Means we did not do it.
Okay. So sorry, I'm saying it's not your fault actually. Normally we do experiments. Zinc doesn't move in the field. So mapping is necessary because there is a lot of heterogeneity. And if there is heterogeneity, we have to spray zinc to make it uniform. Next time it can be improved. But this is a very good finding, very good subject. Thank you very much and very nice presentation. Thank you.
Thank you, sir.
I think disturbance is at Sushma's end. Yeah.
Sushma, you mute.
So may we have the questions from the other members of the judging committee?
Okay, I will come in. Ms. Sushma, presentation was all right, and the work you planned with your supervisor, okay, nano is important, as Dr. Joshi mentions. But if I talk to you honestly, we expect some good science actually from IRA students, even if they are MSc students. So you showed one slide almost at the end, that why you think zinc oxide could be doing better in terms of all those yield seed attributes or quality attributes, right? You remember that slide?
Yes, sir.
So I would have expected you to do that kind of science. You just showed that slide, from the literature and kind of proposal from you that could be the reason wherever zinc oxide has done better in any of those quality parameters, but then you did not do it yourself. I would have expected with even 1 or 2 treatments, you don't have to have 62 treatments, really speaking, maybe I'm wrong, but you could discuss with your supervisor more, that you could have gone a little deeper into the science aspects of it, that why you think zinc oxide, or for that matter, any other nanoparticle is doing better or not doing better, comparatively speaking. So what is your comment on this?
Yes, sir. I need to be taken in the future.
No, like which part of those, different parameters you mentioned in the last slide you think is important, for giving better quality or better seed, emergence, etc. You talked about all.
Yes, sir.
So which 1 or 2 reasons could be there, physiologically speaking, to make the difference?
Yes, sir. The zinc oxide nanoparticle plays a major role. That is, it is a cofactors for many enzymes, like for those enzymes which mobilize the food resource for the radical emergence and all. And it also boosts the antioxidant enzyme activity, which is necessary for the to get rid of seed deterioration.
Okay, anyway, there's some disturbance, I think, rightly so, from your end. Never mind. You can say something if you want to still, in the chat box, please. Yeah.
Okay, sir.
Thank you.
May I now invite Dr. Patra, please?
Ms. Sushma, a very good topic you are working, and you have worked for your MSc thesis in a short time on the nano, really difficult in that way to come out with a clear conclusion. So in that way, congratulations for at least addressing a new area, which is an emerging area, and lot of discussions and prospects we are expecting from that. So in this context, just, since you have worked on this, 3, as I see, zinc oxide, titanium oxide, and silicon oxide you have studied, and what was the purpose of taking these 3 different nano materials? This is one. And second is dry nano zinc oxide you have taken and is shown the best results among all the parameters you have studied. And I understand that you have used these seeds for growing the crops, and you not studied the soil. What is the zinc status? If there is more zinc, available zinc, so what was the purpose of nano zinc oxide use? That should have been thought about in the beginning, of course. So just to know from you, why you 3 different zinc oxide, 3 different nano materials you have used, and have you also compared the nano zinc oxide with normal zinc oxide? What would have been, the differences, the performance of zinc oxide? Because whenever we study, any nanomaterials, so non-nanomaterials also, I think, to be compared with, so that the exact the difference we can understand.
Yes, sir. Thank you for your question, sir. For your first question, that is, these three nanoparticles we have used because these zinc oxide, zinc, titanium or silicon plays a major role in all plant growth and development. And previous literature showed that these zinc oxide and titanium oxide and silicon dioxide played a major role in seed quality and all in other crops, but there was no work on the chickpea crop. That's why we studied this one.
Okay. So in chickpea there was no information, so you studied. And what about the other my queries? Have you with a zinc oxide, normal zinc oxide, I mean to say, have
No,
you
sir.
compared?
We did not do any in comparison with the normal zinc, but in generally the nanoparticles perform better because of its nano size, because of its many properties of its because of nano size when particles.
So, since we are doing the preliminary studies, for really establishing the benefits, we should compare. For establishing the benefits clearly, we should compare. Anyhow, the studies, what you made is new, and the attempts you made very good. So all the best. Congratulations.
Thank you, sir.
I now invite Dr. Patil, please.
Yeah. Now all of us are aware that during couple of decades we have seen the BT, then IT, now Europe nanotechnology. Of course, she has done good work, but as Dr. Patra has already told that she should compare the normal zinc oxide with nano zinc oxide. And I think, Sushma, you may be aware that ICR Sircot Mumbai is having a very good nanotechnology lab, characterization of the particles. There are so many equipments, and how these nanoparticles are distributed on seed, which can be clearly seen through scanning electron microscope, TM, Xrd, as well as there is one good equipment that is atomic force microscope is also available. So I think if you should have sent the samples to Sircot Mumbai lab, you would have got the very good report how it is distributed. Nanoparticles are distributed, evenly distributed or something like that. So I think now you have completed, but in future, if you would like to study, you can send the samples and you can see the how the nanoparticles are playing a role. So these are my suggestions. I think you have not done that, and it should have been easily done, because just you need to send the samples and they could have done the experimentation and the results should have been given to you. So this is my suggestion for future work. Thank you.
Thank you, sir.
So that's up, Dr. Lucha. Yeah. Ms. Sushma, congratulations. Thanks. Very good presentation, very good work. But those were very important inputs have come from Dr. Patra and Dr. Patil and others. But one thing, not in your defense, but one point is there. Punjab soils are deficient in zinc. Even then, we should know the status. So maybe that may. I don't know, but traditional fertilizer and this comparison should have been there. I have 2-3 questions. One, you said potential is, first of all, is uptake is better or any other factor? Uptake from soil by the plant, that's better than the traditional fertilizer?
Sir, can you repeat once again? I could not able to hear.
You said that, not you said, is better than traditional. Is it due to uptake or some other factor?
Sorry, sir, I could not able to hear again.
Uptake of nutrients. Do you hear me? Dr. Nuja, am I audible?
You are audible, sir, but disturbance is at Sushma's end.
So may I repeat again?
Yes, sir, please.
Why nanoparticles are better? Due to uptake by the plant or some other factor?
Yes, sir. Because of its nano size, uptake of.
So it's uptake. Well, anyway,
Yes.
then you said that potential performance has different meaning. Performance is better of the crop or seed treated with nano, or potential performance. Potential performance sort of limit, biological limit. Is that upgraded?
Can you please once again? Sorry, sir.
No. If there is some problem, then okay. Then I will leave. Dr. Patel, if you can help, just respond in chat box, not otherwise. We had very good results in maize field experiments. We put in the package of practices, but in package of practice, my colleague wrote that Terry had they had given us the product that you can go there and use that. So I phoned them and I said, either you come to PAU and sell there. Other point which came up was that that nanoparticles will just they may not be good for our health when we use them. So maybe then we said, okay, we stop it for the time, but we are working on other aspects rather than fertilizers. Maybe in the chat box, if you can, I can get educated. So Shushma, coming back to you, I find I must be wrong. I find that your title is incomplete. I will have written three different or different at least different nanoparticles. I don't know my English. I have studied in Tapra school, in the village school. So to me it looked incomplete. Just think about it. Yeah, thank you very much.
Thank you, sir.
Just a suggestion. We older persons, बूढ़े आदमी जो हैं suggestion देते हैं.
So we'll move on to the.
Please.
We'll move on to the next presentation, which is by Mr. Xrd Kothari from Discipline of Soil Science and Agriculture Chemistry.
Namaste everyone. Welcome to my presentation. Let's start this presentation with a quote of Nikola Tesla. Science is but a perversion of itself unless it has as its ultimate goal the betterment of humanity. In this regard, there are several discoveries in the field of science which have saved millions of lives, which can be divided into different sectors. But which is the sector that had saved maximum number of lives? You may think it as medical field, right? But actually, it is the agricultural sciences that holds the rank for it. Particularly, the innovation of synthetic fertilizers have been attributed to save more than 1 billion of lives. Nearly 40% of nitrogen in our food comes from the fertilizers. In our country, the nitrogenous fertilizers acts as backbone for our food security, as nitrogen is the most efficient nutrient in our soils. In 2020, we consumed nearly 18.8 million tons of nitrogenous fertilizers, in which 80% was applied in the form of urea, and Government of India had spent nearly 50,000 crores as a subsidy on urea. But the nitrogen use efficiency in India ranges from 30 to 40% only. This higher nitrogenous fertilizer consumption with poor nitrogen use efficiency is resulting in the nitrogen leakage to the atmosphere, which ultimately results in problems such as global warming and other associated problems. Also, on the production point of view, we know that the whole world is going through a severe energy crisis, and the fertilizer industry also hardly hit by this crisis. The urea fertilizer recorded its all-time high price in 2021, and which will in turn increases the fertilizer subsidy load on the government. Several countries are already facing the threat to their food security by this fertilizer shortage. Thus, there is emergency need to improve the nitrogen use efficiency of the crop, and the use of slow and control release N fertilizers will be a very smart choice in this regard. So I am Sharad Kotari, representing the Division of Soil Science and Agricultural Chemistry, conducted a study entitled Synthesis and Evaluation of Novel Nitrogenous Fertilizer Products for Enhancing Nitrogen Use Efficiency, with the objectives of to synthesize and characterize novel nitrogenous fertilizer products, then to study the release pattern of nitrogen from these newly synthesized products, and finally to assess the effect of applied nitrogen through these newly synthesized products on yield and uptake of nitrogen by maize. To achieve our first objective, we synthesized two kinds of novel nitrogenous fertilizer products. One set based on nitrogen-loaded nanoclay biopolymer composites and another set based on coated urea products. For NCBPC materials, the nanoclay polymer composite acted as a base material, and NCPC was prepared by graft polymerization technique between acrylic acid and acrylamide in presence of NNMBA as a cross-linker, ammonium persulfate as a free radical initiator, and bentonite as a nanoclay. After preparation, the urea solution was used to load nitrogen in these materials. In case of NCBPC, we replaced 20 and 30% of this synthetic polymer with starch derived from maize and maida flour to get different products. In case of coated urea products, here we have taken different amount of this coating material and clay to get product with different formulations. Here also we had two kinds of coating, one set based on nutshell extract based coating and another set based on naturally polymerizing oil based coating. Then we have characterized our product using different techniques. The X-ray diffraction was carried out to study the crystal nature of these products, where we observed that the characteristic peak of the bentonite was found to be absent in case of both the polymer composites, indicating the complete dispersion of bentonite in the polymer matrix. Also, some of the peculiar peaks present in case of the original starch material was found to be absent in case of corresponding NCBPC derived from them, indicating the participation of starch in the polymerization reaction. The FTIR spectra also further confirm the participation of bentonite and starch in the polymerization reaction. The scanning electron microscopy was conducted to study the surface morphology of these materials, where you observed smooth surface morphology. However, after loading of nitrogen, all the products showed fractured surface morphology with some urea crystal deposition on their surface. During the water absorbency study, the product with 20% replacement had similar water absorbency as that of the original NCPC material. Then coming to characterization of coated urea products, the Xrd pattern revealed that the higher degree of polymerization in case of formulations containing higher amount of this coating material and clay. The same images of oil-based product, we observed that the clay has been deposited on the surface of coating, giving it some rough surface appearance, whereas in case of nutshell-based product, the clay has been penetrated inside the microlayer of coating, giving it that appearance of that mica. Then some EDX revealed that the product had around 30% of nitrogen in them. Then to achieve our second objective, we have carried out incubation study in soil, incubation study in water, and then leaching study, and the better performing products from these experiments were tested in the field. Then coming to incubation study in soil, we have taken 0.05 gram of nitrogen from these newly synthesized products in 20 gram of soil, and we incubated it for 28 days in room temperature. We observed that the maize NCBPC product with 20% replacement had similar nitrogen release pattern as that of the NCPC, and all other treatments recorded significantly lower amount of mineral nitrogen content than that of the urea, indicating their slow release property. During incubation study in water, we observed that the urea has been completely dissolved in water within 7 minutes of incubation, whereas in case of the polymer composite, the release was not even completed up to 30 minutes of incubation. Then coming to coated urea products, we observed that in case of some of the formulations, they had significantly lower amount of nitrogen release during the initial period of incubation and significantly higher amount of nitrogen release during the terminal end of the incubation, indicating their slow release property. Then to study the potential nitrate leaching from these newly synthesized materials, we have conducted leaching study, where we observed that all the products recorded significantly and around 20 to 30% lower nitrate leaching potential than that of the urea. To achieve our third objective, we have conducted field experiment at IRI, and nitrogen was applied through these novel products at 75% and 50% of recommended nitrogen dosage in 3 equal split, one at basal, one at grand growth stage, and another at silking stage. We observed that the 75% N application through novel N fertilizer products resulted in at par grain yield and biological yield as that of the 100% N application through urea. The same trend was also observed in case of plant height and 100 grain weight. And interestingly, the below ground biomass also followed same trend in terms of root morphology. Then coming to total nitrogen uptake, we observed that the 75% N application through maize-based NCBPC product recorded at par total nitrogen uptake as that of the 100% N application through. And the same trend was also observed in case of the grain nitrogen content. Coming to nutrient use efficiency, the 50% N application through these novel products resulted in significantly higher agronomic use efficiency and physiological efficiency than that of other treatments. Then coming to agronomic nitrogen recovery, all the products found to be having at par or significantly higher agronomic nitrogen recovery than that of the urea application. Particularly, we have seen up to 17% improvement in the agronomic nitrogen recovery in some of the treatments. Conclusions. Both bentonite and starch participates in the polymerization reaction during the NCBPC formation. The maize NCBPC product with 20% replacement had similar end release pattern as that of the NCPC, and even at the 75% N dosage, it is found to be equally effective as that of 100% N through urea. Naturally polymerizing oil as well as the nutshell extract can be successfully used to coat the urea fertilizer to impart the slow release property, and the coated urea product 1 found to be very promising in terms of the yield. So at least 25% N dosage can be curtailed without any compromise in the yield. Thus, these novel products can be successfully used as controlled release in fertilizers, permitting more sustainable and high efficient use of fertilizers. Practical implications of my work. We have got approval to file the patent for oil-based product from ITMC IRI, and we are drafting the claims. And nutshell-based coating was an attempt to look beyond the essential oil coating, and it will reduce the cost of coating. And our NCBPC product have implication beyond as a slow release and fertilizer. Path ahead, we have to develop an integrated approach to see these NCBPC materials as in customized fertilizer products by loading more in terms of qualitatively as well as quantitatively. The long-term effect of these materials on soil has to be assessed, and the nutrient use efficiency of the whole cropping system has to be addressed rather than that of a single crop. And we have to go for the smarter production techniques for this production of these materials. Publications. I have got one paper accepted. I have also presented my work in a national conference. I have attended one international conference. I have also participated in the fertilizer orientation course organized by Fertilizer Association of India. I have got Dr. S.P. Rai Chaudhary Gold Medal Award from Delhi chapter of Indian Society of Soil Science for this work. I have awarded the University Gold Medal and other 12 gold medals from University of Agriculture Sciences Bangalore during my graduation. I have cleared UGC JRF in environmental science and other ICR exams. In the end, I would like to acknowledge the help of Advisory Committee and Division of Soil Science and Agriculture Chemistry and ICR IRI during my course of research work. And in the end, I want to conclude my presentation with a century-year-old quote, but it's still relevant now. The fertilizer does no good in heap, but a little spread around works miracle all over. Thank you all.
So may I invite Dr. Patra for having the interaction with the student first?
Yeah, thank you, Mr. Sarath. Compliments, appreciation, I should extend. A very good work you have made in a very short time. As I told in the last presentations, working with Nano and that too in, mostly the MSc work is done in 6 months. So a lot of information you have generated. And this is particularly when we work on nitrogen, so a lot of the scope and there is a need of work. And nano urea by IFCO, we all know, is in the market. So your product also, maybe will get the light to reach the farmers in a time it may take. My queries are, that you are telling that 75% of the new product, the novel product NC BPC is equally good like neem coated urea, 100% neem coated urea, is not it? And that is a really wonderful, 25% replacement is a huge saving for a country. Of course, you have to now go to the field and find out the exact efficiency and other things. In this context, you in one of the slides you are showing that soon after applying this urea, in 7 minutes it is you mean it is hydrolyzed or it is dissolved? What you intended to mention in that? And another thing, have you studied the urea hydrolysis? Because urea hydrolysis and the with the NC and the NC BPC, is it the urea you coated with this material or the nitrogens? So if it is urea and coating with these materials, and have you studied the urea hydrolysis and how much time it is taking? Because your leaching of nitrogens through these different materials is drastically reduced. So definitely the release will be slow in this. So my query here, have you studied the hydrolysis of urea, whether it is neem coated one or with the novel the material coated, whether it is nano or other coating materials? Because that is very, very important an observation which to be made.
For question, sir. For first question, that is urea release you asked, sir. That was for incubation study in water, sir. That means it is not in field. It was completely dissolved in 7 minutes. That means, and other products, the total release was not even completed up to 30 minutes in polymer composite, sir. For second question, that is hydrolysis as direct we have not analyzed, but we have conducted leaching experiment. In NCPC, we used urea solution only, sir. In field also, we have not analyzed the hydrolysis. For that urease has to be analyzed.
Okay. So there are a lot of things to be said, but it is promising. You know, from your MSc work, it looks promising, but you know, since the time is short, better we may discuss later on. Excellent efforts you have made. Hearty congratulations and all the best.
Thank you, sir. It is in 2021 also. It is tested in maize and wheat, sir, and there also we have got similar kind of results, sir.
Any questions from the other members of the judging committee?
Just a small question.
Yes, sir.
Just a small question. What was the purpose for selecting maize crop only? Was there any specific purpose?
No, sir. That means it has been next year we have tested in wheat also. Maize definitely means it is an exhaustive crop and its nitrogen requirement is more also. That's why, and usually they will go for rice only for nutrient use efficiency studies. But you have
tested for wheat also.
That means in next this experiment is going on next year also, sir. That time wheat also tested, sir.
May I now invite Dr. Patil for the interaction?
Yeah, Mr. Sharath, you have done very exhaustive work. And what I find new, that is, it is a new type of work and with new idea. And you have come out with the product, actually that is very important. And you have used all the characterization tools for the nanoparticle characterization, that is SEM, TM, then the surface morphology. And that is last presentation I have given my comment about that. So you have used this one and I think it is very good work. Thank you.
Thank you, sir.
Okay, I take over. I think I am audible. Mr. Kothari, congratulations. Very important, very relevant task, challenging and path-breaking. And I am also happy that you studied root traits, which we normally ignore. And I assume that you have formulated the product, but your advisor must also have played active role, might have thought earlier. So congratulations to your guide also. I could not pick up the name of your guide. Very minor questions. I could not get how much nitrogen use efficiency increases over the normal. I think normal is 20 to 25%, something like this. Normal nitrogen and uptake. 40%.
It is yes, sir. It is around 40. Yeah, 35 to 40. Yeah, 35 to 40% normally we say.
Normally, and in this case,
So maximum we have gone up to
How much is
67%, sir.
Okay.
And
And then why there is so much variation in nitrogen recovery among various treatments? There is a lot of variation. The graph you showed. Any
sir,
reason?
dose was different, sir. That means in some it was 75%. Both each product 2 dose we have tried, sir. 75% and 50%. 75% high, but 50% was higher, sir, due to that lower dose. And 75% it was.
If I could understand it properly, nitrogen use efficiency was not that much variable as nitrogen recovery. If I could understand, if I'm correct. So that is why I'm asking why there is more variation compared to nitrogen use efficiency. Anyway, coming to a critical point, I will say outstanding work, everything outstanding, again, publication is not matching.
Sir, because we are going for patent, so we have
Okay,
prepared two products.
Oh, you have very valid point. You have very valid point. Thank you. I should not ask this question. I should not ask. Thank you. Thank you, Dr. Lucha.
Thank you, sir.
Thank you all.
So now we move on to the next presentation, that is by Ms. Geeta from the Division of Vegetable Science.
Honorable Chairman of the session, respected jury members, my dear friends, good evening to all of you. Nothing will benefit human health and increase the chances of survival of life on earth as much as the evolution to the vegetarian diet. India is a poor performer in terms of global hunger index, where it impacts on development of a country. India stands in 101 position in global hunger index 2021. Among hunger, hidden hunger is the serious issue which is due to deficiency of micronutrients. It often linked with malnutrition. This malnutrition shows its effects into stunting, wasting, and obesity. Though these mineral nutrients do not provide any energy to the body, but they are absolutely necessary for various metabolic activities. Along with micronutrients, antioxidants are the most important health beneficiary elements that are the scavengers of the free radicals produced in our body. Our major diet portion is contributed by the vegetables. These vegetables are the source of mineral nutrients, antioxidants, and vitamins. India has emerged as second largest producer of vegetables with 196.2 metric tons. Cauliflower is most grown brassica vegetable in India. India ranks second in cauliflower production in world. It is a rich source of dietary minerals, protein, dietary fiber, and vitamins. Presence of self-incompatibility and male sterility has made it possible to develop cauliflower hybrids in terms of uniform maturity and better curd quality. Studies on quality traits is restricted to snowball cauliflower. We can find almost nil reports on the quality traits in Indian cauliflower in terms of exploitation of heterosis and identification of good combiners. Therefore, there is a need to study heterosis for quality traits and identification of good combiners to develop nutritionally rich hybrids. Glucosinolates are secondary metabolites found in cauliflower which confers biotic resistance and health benefits. We do not find any information on expression pattern of glucosinolate pathway genes in Indian cauliflower. In order to fulfill this gap, we have postulated our thesis title as Studies on Heterosis for Quality Traits in Indian Cauliflower. Myself, Geeta, from Division of Vegetable Science. The objectives of my study are to study combining ability and heterosis for yield and quality traits, and to study expression pattern of major genes involved in glucosinolate pathway in cauliflower. For the first objective, we have taken 10 CMS lines, 5 testers, along with their 50 F1 hybrids from early group. From mid-maturity group, 5 CMS lines, 4 testers, along with their 20 F1 hybrids have been selected. Following morphological parameters have been recorded. From each F1 hybrid, line and tester, we analyzed 9 mineral nutrients, 4 antioxidants, and 3 individual glucosinolates. Stepping towards results, hybrids performed better than the parents. Marketable curd yield and net curd yield of the hybrids is quite more than the lines and testers. For these agromorphological traits, hybrids have shown wide range of heterosis over mid-parent and better parent. The dietary mineral content of the F1 hybrids for iron and zinc content was higher than their parents. In mid-maturity group, zinc and sodium concentration was found to be higher than their parents. For these dietary minerals, we observed low to high concentration of the content in cauliflower curds. F1 hybrids of both maturity groups have exhibited a wide range of heterosis in both positive and negative direction. We identified 2 lines and 1 tester from early group, 2 lines, 2 testers from mid group as a good combiners for the mineral elements. Content of glucosinolates and antioxidants in F1 hybrids was quite low as compared to their parents, except cinegreen content in F1 hybrids of early group and ascorbic acid content in mid-maturity group. Genotypes have shown low to high concentration of antioxidants and glucosinolates. We observed heterosis over mid-parent and better parent for these glucosinolates and antioxidant in both direction in both maturity groups. CMS 84415 line was identified as good combiner for the glucosinolates. The line N2982 and 394415 were identified as good combiners for both antioxidants and glucosinolates. In testers, DC415 and DC71 were identified as good combiners for both antioxidants and glucosinolates. In mid-maturity group, line CMS 8401 and tester DC309 were identified as a good combiners for both antioxidants and glucosinolates. Stepping towards second objective, glucosinolates are the secondary metabolites found in cauliflower. When they comes in contact with the myrosinase enzyme, they produce variety of compounds. Among them, isothiocyanates are the most common products, and they confers resistance to biotic stress, imparts flavor and pungent smell to the cole crops. They are the strong antioxidants and prevent carcinogenesis. Evolution of tropical cauliflower from European type cauliflower in Indian condition has made genetic changes that has also impacted on changes on glucosinolates also. Therefore, there is need to study these changes over glucosinolates in Indian cauliflower. For second objective, we have taken 9 commercial varieties representing all maturity groups. The varieties have exhibited predominance of the glucoerosine content as compared to cinegreen and progoitrine. The glucoerosine was predominant compound in early, mid-late and late groups, whereas in mid-early group, progoitrine was higher as compared to glucoerosine and cinegreen concentration. 14 PCR-based DNA markers from glucosinolate pathway genes were synthesized and screened in 10 commercial varieties. 10 primers were found to be polymorphic in nature. 2 primers, namely MYB29 and GER1, did not produce any amplicon, while MYBA09 and MYBA03 were found to be monomorphic. To study expression pattern of glucosinolate pathway genes, 8 major aliphatic glucosinolate genes were selected. The results were expressed as a relative fold change over the base variety Pusa dipali. Pusa paushija has the highest relative fold change of 2 genes, MYB28-1 BOGSL elong. Pusa ashwini has the highest relative fold change of gene MYB28-3, and Pusa snowball 1 has highest relative fold change of CYP79F1. BRAOP22 gene has highest relative fold change in Pusa snowball 1. MYB28 to MYB29 2 genes have higher relative fold change in Pusa paushija, and MYB29 1 has highest relative fold change in Pusa ashwini. We have observed positive correlation between the expression level of the glucosinolate genes and glucosinolate's content. The positive correlation was found between MYB28-2 and AOP2, MYB28-1 and MYB28-2, and MYB29-2 and AOP22. Identified best combiner lines and testers can be utilized for breeding of nutritionally rich cauliflower hybrids. The superior F1 combinations can be directly used as F1 hybrids. The information on markers and genes from the glucosinolate pathway will be helpful in further studies on this compound. I would like to acknowledge Chairman and Advisory Committee members, Head and Professor, Division of Vegetable Science, Division of Genetics and Agricultural Chemicals. Thank you one and all.
No hands raised as yet from the members of the judging committee.
Why so? जोशी साहब हाँ आगे सभी आगे
Okay, because nobody is asking, so and her name is Geeta, I think.
Yes, sir.
Oh, very good name,
Geeta
you can speak hours and hours so we can listen. Good work, good presentation. This is challenging subject. See, working on vegetables is not easy. Very difficult, and when you produce habits and you have different lines and different traits, it's not easy. So give me just one answer. How your research will help farmers? Sorry.
Yes,
Got
sir.
my point? Sorry, you are too young to answer this, but think, from today you have to start thinking big.
Yes, sir.
Yeah.
Sir, if farmers got to know about the nutritional status of the crops, so they might have take this the identified lines or testers or the superior hybrid combinations for their cultivation, sir.
Good. This is how you have to answer. Thank you very much. I am done, Dr. Dhillo. Thank you.
No, there are other colleagues, my dear. Dr. Patra
Dr.
Saab.
Patra, please.
I thought also to ask something. You were on several nutrients, about
Yes.
9 nutrients, and you have looked into where the promising combiners for multiple minerals you have identified, and my query is here in the perspective of soil fertility. In a scenario when these nutrients are in deficient in the soil, what will be the performance? For example, zinc, widespread deficiency in the Indian soils, about 40% soils deficient. So also sulfur and other nutrients. How these lines, what you identified, will be efficient to express its the traits. You understood my query?
Yes, sir. Yes, sir. I understood,
Ms.
sir.
Geeta, otherwise it is wonderful presentation and lot of perfect implications are there. A very good study. Just this is my query. I thought something, let me ask also.
Yes, sir. Sir, actually, we didn't study about the gene interaction, whether it is due to dominance or like that. So, and also we did not analyze any soil nutrient status also.
But then how you identified what is the 9 different elements you are talking, the combining it, whether you applied, whether it is an optimal dose, what is the status, and then when the soil is in that way, no deficiencies there, then you study, then you will get a clear picture. I don't, because since it is not my actual subject, but what my lateral thinking about the your presentation.
Yeah.
May I now invite Dr. Bansal for the interaction?
Okay, Ms. Geeta,
Yes,
how do
sir.
you measure the expression of various genes?
Sir, the relative fold chain.
No, relative fold change is fine, but how did you, which technology did you use to measure the level of gene expression? Which method? Which instrument?
Sir, RT-PCR.
Which RT-PCR?
This
RT-PCR,
QRT-PCR.
semi-quantitative RT-PCR.
QRT-PCR, sir.
What is QRT-PCR?
That quantitative
How does it differ from the semi-quantitative PCR, RT-PCR?
Sorry, sir, I don't know the difference.
Students, actually speaking, should be looking into all the aspects much more seriously at this stage now than later.
Yes.
One. Second, out of so many different genes you studied the expression of, and you found a positive correlation with glucosinolate content, right?
Yes, sir.
So could you say, out of so many certain different genes expression studied, was there any one or two which are more critical in terms of their expression that they must express first in the pathway? Was there any kind of information available in the literature otherwise or from your studies?
Sir, I'm not getting, sir.
Okay, no problems. Don't worry about it. Take it easy. But you must interact with some, friends who are otherwise molecular biologists, from the division. That's an advantage of IRI. We'll talk about it in general status when we think about more. Thank you, Mr. All the best.
Thank you, sir.
May we now have the questions from Director, sir?
Yes, Geeta, thank you for excellent presentation. The mustard breeders, they are trying to breed away the glucosinolates, and you said you want to enhance glucosinolates.
Yes, sir.
So there is a contradiction in the two approach. Why you want to bring in glucosinolates as such, they are not harmful, they are not good for health. And the other question I have is that there is a lot detail known about the genetic control of glucosinolates. It's a transcription factor gene MIP32, which is well characterized. And if you just knock down this, you get the glucosinolate-free lines in mustard. So managing at that level for reduction is fine, but you wanted to increase the glucosinolate content. Any specific regions can you cite?
Sir, especially we want to increase the content of cinegrine and glucoerosine, whereas this progoitrine is anti-nutritional. It causes goiter. So the lines which lines and testers which we have identified, they are having lower heterosis for the progoitrine and low combining ability for progoitrine and higher combining ability for glucoerosine and cinegrine content, sir.
Okay, it has to be seen at the genic level. I mean, then we can go for combining ability and heterosis, because heterosis combining ability will come from the gene. And these genes are well understood, in fact, at molecular level. So I think some molecular analysis of the material that you are handling would be really giving a very good direction. Thank you.
Thank you, sir.
Thank you, sir.
Yeah, I will chip in. Dr. A.K. Singh, just for sake of exchanging idea, we have a variety, canola variety, gobi sarson, PGS7, doing very well. So we invited the industrialist. And one of the industrialists, I think probably from Moga, he met me later on and he said that we want something glucosinate and we will purchase whatever is available. We are purchasing normal sarson from Rajasthan. And he was telling many advantages of glucosinate. Of course, he is working extensively on quality in mustard at the cost of yield also. But anyway, coming back to Geeta, very good presentation. And just when I was at IRA, my roommate was working on cauliflower. So I went to those dates. I'm not going to ask any question. in relation to science. Please name 3 outstanding scientists of IRE who worked in cauliflower.
So one is Dr. Pritam Kalia, sir.
Then I asked 3.
Sir, one of my were guide.
I hope Dr. Kalia must also be listening. Does he agree?
No, sir, he's not there. He is retired.
He's not there?
No, sir. No, sir.
Bete, I don't know how to rank them, but Dr. Vishnu Swarup,
Yes, sir.
Dr. Chatterjee, Chaudhary, they were the stalwarts. And sometimes I feel sorry that we asked names of the Director General of ICR. Some candidates are not able to even name Dr. Swaminathan. They are the persons who did it. So, Dr. Pritam Kalia is doing wonderful. I have interacted many times with him, very good friend of mine. But I think maybe somebody from vegetable throw more light. I think I will put Dr. Vishnu's group number 1.
Yes, sir.
No, I mean somebody from.
And Dr. Gill is also there. Dr. H.S. Gill, sir.
Oh, Dr. Gill worked for only for some time.
Sir, quite for a long time.
Don't try to hijack me with turban.
Katran station.
No. Dr. Gill doesn't come to the level of. Let me say that straightforward. We used to take lunch and dinner together in Jewel Box mess, JB mess. Anyway, he was also outstanding, but stalwarts were. Anyway, that's what Anyway, let us move on. I think one person is left. What is his name?
No,
Shubha.
all the presentations are over, sir, but
Shubha.
only one presentation which was not completed. So the student has shown her inability to make the live presentation. So can we run the pre-recorded presentation again, and then we can judge on that basis?
That depends on IRE authorities.
But
we can do that. In fact, the candidate was contacted. Dr. Rasmi, what did
yes,
she
sir.
say?
Yes, sir. I contacted her, but she said she won't be able to join, even for question answer session.
Okay.
Yeah, she's in a very far off location and she's not getting the connection also. So she said she
So
wants
can
to
we- can
abstain.
we-
So very
But she
good
wants to
example of
abstain. She does not want to participate, sir.
very good example of digital divide.
We can run through the presentation, Dr. Because she is traveling, no? She will not be
So
here.
tomorrow morning she is traveling, sir. Today she is there only. But
No,
she
but-
says now she doesn't want to appear and
Okay.
let my presentation also not to be played. That's what
Okay. All right.
after repeated convincing, she was not ready, sir. That's what.
All right.
Still, her chairman may be requested.
Vishnu sir,
Yeah. We still
You can-
open for her.
Yes.
No, I have nobody to decide. This is to be decided by IRE authority. I am not naming anybody.
No, sir. Chairman of the student. Chairman of.
Okay. Yeah.
What? The
No,
chairman
I
of-
think
Chairman,
Yeah.
can contact her again or.
And communicate to us accordingly.
Yes, sir. Some communication written or something has to come. I told her to mail us or send me a WhatsApp message, but I have not received, sir.
Okay, madam. Yeah. Dahuja.
Yes, sir. So with this, we come to the end of all the 22 MSc presentations, sir. So as I said in the morning, so total we have 36 presentations, 22 from MSc and 14 from the PhD. So presentation of the MSc students, they were scheduled for today, and tomorrow we have the presentations of the PhD students. So that means 14 presentations will be held tomorrow. So would you like to, sir, give some concluding remarks today, just briefly?
I have been giving,
on the presentations of the MSc students.
No, I have been talking too much in between. But my point is that can we finalize this, because this will be independent of other one.
Yes, sir. Yes.
There is no interference, there is no interaction, there is no epistasis.
Yes, sir.
So if we can just finalize.
I would like
this.
to request, sir, all the jury members. I will thank all the jury members and the chairperson, and would like to request that, sir, if possible today evening, sir, kindly finalize.
Yeah, let us now it is in our mind. Or गल को तो भूल
Can
जाएंगे
I-
मेरे-
can I-
Sir, अभी बस शाम तक
हाँ जी,
आप-
हाँ, Dr. Mansur.
Yes, sir.
Can I make some general remarks?
Yes, sir.
Can I make?
Yes, yes, sir.
Sure,
Please go ahead.
Yes,
Yeah.
sir. Yes.
sure,
Can I
sure,
make?
sure.
Yeah. Okay. No, actually, I just wanted to make a few observations. While the students have worked very hard, and try to make very good presentations, but in certain cases, I think there is a requirement at the chairman's level or the level of the advisory committee to do kind of a rehearsal in a competition like this, before they record their presentation. I'm sure they would have done it, but nevertheless, I think it's important to still come to kind of a perfection, before these presentations are recorded. Number one. Number two, as has already been mentioned by the chair, Dr. Dhillon, that these kind of statements and, messages from some of those key personalities really not required. I mean, this is a personal observation as well. I don't know how to convey to the students or to the chairpersons. We should not be really speaking, interfering too much. It's a freedom of their own speech and expression, but it takes away few extra minutes actually speaking, before they come to their title of the presentation. That's second. And third is, I think some kind of an interdisciplinary nature of work, to be reflected in these presentations is very important. I know it is only for master students. They've got only about 6 months at the most to do their thesis, but nevertheless, the beauty of IRI is that we have all those 26 disciplines under one roof. I think that kind of interaction, therefore we have this advisory committee, to also be reflected in case, because several of these presentations, and to be very honestly talking about presentations from horticulture or fruits, seed science, vegetables, floriculture, environmental science, they must have some kind of input or interaction from disciplines, basic disciplines like plant physiology, biochemistry, genetics, and plant breeding. Because they are also doing, while the subjects are classified as horticultural subjects, I mentioned before vegetable floriculture, but the science part of it is either they're doing physiology, or they're doing biochemistry or processing quality, or they're doing molecular biology. At the last presentation of vegetable science, we saw some of that kind, or they're doing genetics and plant breeding. So I think a very good support or system is required for all these MSc students coming from these disciplines, and could be one or two more which I would have missed. I think that kind of interaction is really a must. There's something to talk about it. And the last point I would want to make is, while the path ahead is important, but students must also be prepared, really speaking, to see that what is something realistic in terms of not necessarily that we look forward to some kind of, recommendations coming out of MSc work, but that should be more of realistic and practical in nature, than otherwise. And last but not the least is with regard to their publications. I can understand that publication probably would attract some marks, but most of them, in fact, had their publications in NAS factor, NAS rating less than 6. That means they did not have any international impact factor allotted to those journals. And I think there's some kind of a pressure, I can understand, on MSc students to publish, but therefore, can we think of a mechanism how to deal with this, in subsequent years, of really speaking, doing away, with kind of, system, so that they can really add, as one of the, I think, expert members, or Dr. I.P. Abral mentioned, to add into the whole mandate. That should be the objective, I believe, rather than for MSc students, rather than going for immediately a publication. I'm not against Indian journals, but publication in journals which are less reputed, and we should encourage, to have a kind of international scene, because we are talking of IRI. IRI is supposed to be, giving a direction to rest of the country in terms of, research. So our MSc students have been grabbing very good opportunities, I know, in national, international fora. So therefore, it's important to set an example, even with regard to the publications of MSc. I'm sure tomorrow PhD students will do, certainly, no doubt, a great job. I have heard them before, and I've seen the kind of progress which is being made. But again, I would, of course, congratulate all the MSc students for their hard work, and some of the students were outstanding, So I think once we are judging them, I think that becomes very comparatively, kind of, a judgment, that they stand out. from the same institution. So I think they could learn from their own friends, and there should be more of interculture interaction among students themselves, and kind of scientific aspects, scientific questions will be more important than talking about, whether something was sprayed or something was provided to the soil and some parameters changed. But why? That question they must try to answer from the scientific point of view. I think for a MSc student, even one small question, one small query would be enough, from innovation point of view, rather than going into, because we are now come of age at even an IRI, long time to contribute it, really speaking to the basic understanding of any single given problem. Let it be, in fact, one small experiment, only one research paper, I would say would be even enough for that matter for MSc students. So anyway, these are some observations. Science must prevail. I think it's important, and we have all together to play a big role. And particularly, I'm excited to see the leadership of our Honorable Director, Dr. Ashok Singh, who himself is a well-known geneticist and plant breeder and molecular biologist and biotechnologist. So I think we expect more, from this leadership, including Dr. Rashmi Agrawal as the Dean, and several head of the divisions, doing excellent piece of work. So I think together we must try to make a difference and bring up even those disciplines which are otherwise not able to do as much comparative speaking compared to many others doing better. With this, thank you for this opportunity. And these are my few observations.
Thank you, sir, for the insightful comments.
We can ask other members also if they want to say something.
Dr. Joshi, sir?
Nothing.
If they want to say something tomorrow.
Okay.
No, only one thing. Thank you very much. It was a beautiful day. The future of country is very bright. Please see, Ashokji and Dhilanji, it was very difficult for me to score, actually, somewhere neck to neck. So actually, I was very much worried, how to score. And future is bright. And Dr. Bansal, just wait for 20 years. These 22 people will be sitting on top of different places. Many of them. Thank you very much, Ashokji. Thank you. Thank you, Dr. Dhillon.
So I was thinking to not to say anything, but there are 2 points which I think probably if IER authorities want to maybe implement it tomorrow, so because of them, I will just take 2 minutes. First thing is, I'm very happy that most of the candidates are girls. So India is making progress. And very difficult, Dr. Joshi, same thing happened earlier also. I told you, best thing is that this is the best certificate that I started in PU, instituted 5 gold medals. Could not do this because of COVID, but the decision is taken, money is kept. Then choice of the project is always relevant, very, very important. And of course, sometimes they have gone in depth, sometimes they have not gone in depth. Then Dr. Bansal, it is not 6 months. It depends upon the mentor in MSc. Mentor is much more important in PhD. I conducted research I joined MS in 1967 and I generated 2-year data at that time. So it is not 3 months, 6 months. The advisor is there to help the student to generate more data. And we work as a group. Then I also feel very happy to see so many awards. The students, they are working as total personality, unlike us. We were just studying in क्या कहते हैं nose की direction में सीधा आगे. No, didn't think of anything else. My maternal uncle gave me ₹80 to give examination for IAS. I spent those ₹80 in Vivek and Payal and Liberty Theatre. Didn't submit my application because I felt very happy. Then now see the certificate, extracurricular activities, etc. So wonderful experience. And number of participants also, 500 participants. Now 2 points, Dr. Ek Singh, why we are compelled. I didn't want to say anything because I was just giving my input in between. 2 points. Number 1 is, can we say tomorrow, the first, in fact, I liked only one quotation at the end about Indian society being vegetarian. That was to the point. Otherwise, starting with medicine versus agriculture, etc., just when we have limited time. And again, acknowledgement. We again spend some time, I will not say waste your time. We again spend time. If there is something which is other than director of IRE, etc., or parents or friends, may say a sentence, something like that. So that's one point which I wanted to say. Second time, many times I felt that I am not doing justice with the candidate. I'm asking a question which is not supposed to be asked from. So maybe from next year, if not tomorrow, if advisor can also be there in the discussion, because it's a learning experience for me. Let me say it frankly. I have noted some points which I will be telling a PU. I am sharing with you and we discuss on telephone. I will share with them also. So these are 2 points why I thought, okay, let me share these 2 points. But on the whole, wonderful experience, very difficult. And since it is difficult, Dr. Joshi, difficult things must be done immediately. So let us select 5 or 6, whatever is to be done. Provided IARE authority agree to that.
So one,
yes, sir, we are.
Yeah.
No, it is not question of laughing, Dr. Joshi. I tell you one experience. In one of the meetings, one of my mentor was vice chancellor at that meeting, and he became in the plenary session. I said, okay, he will chair it. And he hijacked that. And when I came back, I was ADG, Dr. Mangala was GDG. He got, I learned many things from Dr. Mangala and Dr. Prodasab. He said, Tillo, never let the old person hijack the proceedings. Always, I say DDG or ADG should be the chairman in plenary session. So here we will go by the advice or the direction of Dr. A.K. Singh and other persons of IRE. Otherwise, old persons, we don't know much. I mean, I don't know much, and I will hijack the.
This is 100%. This is 100% the prerogative of the judging committee. Absolutely nothing to be done from the IRI side.
I mean, as far as should we do it today or do it tomorrow? I want.
No, I think if it can be finalized today for the MSc student, that would be better because
because it is in mind.
We'll have that will leave less.
But Dr. Bansal wanted to say something.
No, I thought since we are talking of only the evaluation of 30 marks out of 100.
Yeah.
So therefore, therefore, what I have done, I have prepared a sheet, of these marks, which I could, send it across, through WhatsApp after taking a picture, because then it has to be total with, with 100 marks.
Yeah.
So we cannot say at our end 1, 2, 3, 4, 5.
Out of 30 marks, let us identify 5, 6, 7, 8, 9, 10, whatever they want. Let us identify that.
No,
who's on number 1 in your list? I mean, is there only now? I think others should leave, please. Only Dr. A.K. Singh, Dr. Those who are to evaluate. Dr. Rashmi.
Yes, sir.
And